It's straight up A. And other folks will pop in. So we'll start with an intro, just general intro.
Great.
Give me like 3 minutes.
Okay, we're alive now.
Awesome. Well, welcome to Magnitude Live episode 10. I'm Ted, and I'm here with Tony from Magnitude and Gil from Japan. Yay. Thank you, Gil, for joining us. And it's midnight?
It is. It is exactly midnight.
Bing. All right. So witching hour. It's tomorrow. It's yesterday for him, for us, something like that. And we're going to have some more guests jumping on. What is Magnitude Live? Well, most of us have been stuck at home for a number of weeks, whether we're teachers or parents with kids at home, and we're looking for super fun things to do. Magnitude puts stuff in space, specifically experiments aboard the International Space Station. And we've got one coming up, and we need your help. So Exolab 8, our 8th mission. Hey, Shivar. Shivar's just joined us from South Africa. Great to see you, man.
Likewise, Ted. It's good to see you.
Cool.
Good evening.
Yeah, we basically setting these experiments up to the space station. And in this case, we need your help. And we're getting help from students around the world. Shivar works with some students in South Africa. Gail's got students in Japan. We've got tons of students here in the United States, in North America, in Canada and Mexico. My goodness. And they're speaking of Mexico. Javier has just joined us from Houston, and he works really quite directly with a lot of South American and Central American countries. Great to see you, Javier.
Hey guys.
And so these experiments, and this one, we're trying to find the right plant to send to space on our next mission. And we're looking for this symbiotic relationship between a bacteria and these plants, these legumes, which you might normally think of as beans, but those also include clover and alfalfa and others. These legumes really need this relationship with the bacteria in order to produce successful crop. The bacteria provides the nitrogen that the plants need, and through photosynthesis, the plants provide the sugars to those bacteria, which they dearly love, and they really join things together. And now, to another big drum roll, we have Dr. Richard Parker from the Gilroy Lab out of Wisconsin. Nice and toasty there this morning.
It's funny.
Good deal. And so that's what we're doing, and we need your help. And so over the next several weeks, we're going to be thinking about these different legumes. But today, Gil from Japan is going to be talking about the effects that gravity has on life and what happens when we remove that vector from the equation, something that evolution has relied on forever, at least here on Earth, as far as I know. And I'm going to turn it over to Gil. And we've got other guests coming on the show. In about an hour, we're going to have the Pawns system, which is a device that actually goes in the veggie lab where the astronauts grow their vegetables and what have you. And that's made by Tupperware. So David Kasuma is going to join us at 10. And we've got some culinary interests going on with Leguminaut from Orlando. And then at 11, we're going to do, we normally do some kind of activity that's not directly related to the ExoLab, the experiments that go to the space station. And we're going to be launching another mid-altitude balloon, this time from Southern California with Liam Kennedy, part of ISS Above. And they're going to be working with the YWCA and a bunch of young gals there that are learning about these missions, and they'll be sending that up between 11 and noon. Oh, and look, we have Mr. Wilkinson. Good to see you.
Good morning. Sounds like another full program, Tony. Ted.
I'm going to turn it over to you, Gil, and it's all yours, man.
Thank you, Ted. Hi, everyone. My name is Gil Cawthorn. I live, work, research, habitate in Osaka, Japan. And I wear a few different hats. One of them is I'm an Astrobotany Research and Education Program Coordinator in Osaka. I am also one of the PIs, the primary investigators here with Magnitude, which is super fun on the next launch. And today, I'm going to be talking to you guys about gravitropism, the effects that gravity has on plant life, and how you could actually do a very, really simple experiment at home to see this with your students, with your kids, with yourself. I do that all the time, but I'm just kind of weird that way. So we're basically just going to talk about first a preface of why this matters. What is astrobotany? Why do we care about plants in space? And then the effects that living in space has on life and how that connects to gravity. So I'm going to try to share my screen here in a second. Bear with me because I got some
Oh,
slides.
I suddenly heard Jim McKay spanning the globe. We've got Germany and Canada and Japan and South Africa. Spanning the globe to bring you the best astrobotany possible.
All right. Let's see. Are we good here?
Yeah.
Present. Let's see.
Who's the artist? Who's the artist? The character there? The Astro Farmer?
That was actually Kai from Astrobotany.
Oh, that's Kai. Oh, cool.
I did that. Yeah, I love
so
that.
I saw a piece of his last week.
Yeah, I wear my Astrobotany shirt weekly. I love it, and that's one of his illustrations. So if you guys don't know, go to astrobotany.com. There's a lot of really great educational materials there and a lot of cool artwork. So check it out. Right, so let's get started. Astrobotany, education, and gravitropism. That's what we're going to be talking about today. So it's a topic that I love, that's very dear to my heart because it's important and it's weird. So like I said, a quick preface, like what is astrobotany? Why does this matter? Why should you care? So very simply, astrobotany is a study of plant life and plant interactions in space environments. So how plants grow in space, how they change in space, how the space environment stresses them out, how the overall biology of plants, how it changes and how it reacts to space, and the development of systems for better plant production in space. It's a cross-disciplinary field. It isn't just botany. It isn't just space science. There's engineering involved. There's chemistry involved. It draws from a lot of different fields. And what's really cool is how it doesn't just apply to space science. And we're going to see that as we go along. So astrobotany is a study of plants in space. Okay. And Dr. Barker, is there anything you'd like to say about that image on the right?
Just that, well, that's an astronaut harvesting the first Arabidopsis plants to be grown in the advanced plant habitat. So that's made by Sierra Nevada Corp in Madison, Wisconsin. It's the biggest, most complex plant growth system ever put on the International Space Station. It's got cameras, lights, CO2 scrubbers. It's a really impressive, noisy piece of equipment. Those plants, they were harvested, they were put into the -80 freezer after being fixed with concentrated ammonium sulfate solution, a chemical called RNA later, and essentially preserved the samples, brought them back down to Earth. And currently they're being researched by Norman Lewis and his team. And what they are doing is they're sequencing the messenger RNA to look at all the genes that are being expressed, and they're also looking at the metabolites. And this is what makes this experiment so special. They're looking at how the metabolic components of the cell are changing, the chemistry inside it. So that's something that will be published in a few years from now, maybe hopefully next year, but it's really exciting. But the best way to understand spaceflight environment and the best tool that we have at our disposal is Arabidopsis. And that's what you can see front and center in the center of the Arabidopsis. But it has many relatives, and Mizuno is one of those. Over to you, mate. Oh, and on the right, the time lapse. Okay, great. One last little thing to add. So we can look at a plant like Arabidopsis here growing in lovely green. We can measure things like how much green area there is or the shape of the root system. But one of the really awesome new tools that we have at our disposal is time-lapse photography and looking at how things change over time. And that's what you see on the right there. That's a wild-type corn variety, and that'll hopefully be launched on the next SpaceX if COVID doesn't get in the way. And you can see, if you look at a few different time points, you can see different characteristics of its root system architecture. Its primary root growing and its lateral roots coming out the top. Many interesting things to measure. Lots of these things regulated by gravity.
And Dr.
Natural
Barker,
blood-
that looks like those look like magenta boxes. Is that right? 3 of them?
Exactly that, yeah. Customized.
And what's the media?
Mainly agar. So I had to slice the bottom of the magenta cube off. Took a few attempts to get it right, and then you had to do it hundreds of times to get enough to use for large-scale experiments. But it works. It's then sealed up with an aquarium gel. It's the labeling as well. There's so many different things you have to take into account, like the type of sellotape that you use. NASA always has to use Kapton tape. It's like this kind of clear plastic stuff. And then you have to make sure the labeling is clear as possible. So if the astronauts have to interact with it, there's no superfluous information. You have to go through a lot of iterations and overthink things. And then it's just agar inside. We tried a range of different medias, and then went back to our favorite. But again, that's part of the game. But not everything has to go up. We can learn a lot of things about plants and how they will interact with gravity by just growing them on Earth. And I think that's the focus of this talk. What's the best assay that Darwin invented? I think gravitropism.
Yeah, and this, everything that he just said just ties back into this broad developing field, which is astrobotany. It's something that, what's exciting about astrobotany is that there's so much that just isn't known. There's so much that is yet to be discovered, and so much that we have to continue pushing forward with, not only for our development in space, but for our development here on Earth. So why is it important for future space missions? It's a critical component for long-term space habitation. Obviously, there's like the food part of it, right? We can see, let's see, we got, by the way, on the screen, are you guys seeing
Scott Kelly harnessing the zinnia flowers? That must be Valentine's Day.
Yep, there we go. Okay. So there goes astronaut Scott Kelly, and those are flowers. Those, as you can see, he's not going to eat those, but it provided something almost as significant as nutrition to him and to the other astronauts that were taking care of the plants during that mission, which was a connection back home. Taking care of plants during space missions has shown to mitigate a lot of the psychological stresses that come with space-induced quarantine, which is something that we can kind of relate to a little bit right now. But studies have shown that astronauts that have taken care of plants on various missions have performed better in certain tests regarding mental health, regarding stress levels, all that kind of stuff. So that's super important. That also, the other part of it is, the nutrition. We need to have sustainable food production on Mars, because we are not going to be able to send food over there, or vice versa. Plants on space missions also help with gas cycling, oxygen and carbon, and they help develop self-sustaining bioregenerative systems. We need systems that are able to develop systems that make life better for those that are habitating in space.
Well, yeah, and it kept him in good stead with Amiko, his wife, spending an entire year away from her. He can still give her some flowers on Valentine's Day.
Yeah, so it's important. It's a critical part of it, and it enriches the lives of those that are carrying out the research while they're up there.
And apparently you can eat zinnias. There is a zinnia cookbook on the internet somewhere. But
really?
But people don't tend to do it. It's a weird thing to do, FYI.
Let me see. Okay, here we go. So that's all cool. So usually when I've talked to people about astrobotany, they're like, oh, like the Martian. It's like, well, yeah, kind of. But then it kind of goes into this question into why does it matter? Like why? Cool. So we can grow a couple plants around the Earth and on orbit. That's really expensive. So why does that matter? Plant life support systems that are going to be used on Mars, on the moon, or on orbit need to be energy efficient, produce nutrient dense yields, produce minimal to no waste, have to be reusable, resilient, cost efficient, sustainable, and self-reliant for the most part. They need to be able to work and produce in some of the worst conditions ever known. Now, if they can do their job sustainably, faithfully on Mars, they can do it here. And we have last time I checked, we have a lot of people here on Earth that are suffering from hunger. And there's a lot that we have done as a species to the environment in our food production. So whatever we figure out moving forward that could be used in space could absolutely be applied here to alleviate the suffering of hundreds of thousands of people around the world that don't have enough to eat, that live in places where right now, because of climate change, either have too much water or too little water, can't really produce food on a regular basis. That research that we're all taking part in right now will contribute to not only the future of spaceflight, but the future of Earth-based agriculture, which is super important. I love space. I grew up next to Kennedy Space Center. I love it. But the space part gets me excited. The part of helping people here on Earth, to me, is even bigger. We have to get this right. We have to get this right. To be able to move forward as a species, we've got to get this right. So this is why astrobotany is important here on Earth. And this is why studying gravity, studying these symbiotic relationships between rhizobium and legumes is so important, because they're all components to a bigger picture, okay? Sorry, I could go down this rabbit hole for a long time, so I'm gonna move on. I apologize.
No, that's absolutely fine. That should be neat. Is a little shout out in Japanese too, just for adding a little international flavor to it, if you will.
Well, I mean, everyone that's listening right now, a quick Japanese lesson. Sorry, one of my daughters just zombied down the hallway. Astrobotany in Japanese is a fun word. It's Uchu Shokubutsu Gaku. So if you're listening, if you want to try it, it's Uchu Shokubutsu Gaku. Uchu means space. Shokubutsu Gaku means the study of plants. So it's literally, if you just read the kanji, it's space study of plants. So next time, if you want to impress someone after getting some sake and sushi, be like, hey, you know what? Uchu Shokubutsu Gaku. It means astrobotany. You're probably not going to ever use it in conversation, but now you know, okay? So anyway, what are the challenges to life in space, okay? So terrestrial life encounters so many stressors in space. We, over millions and millions and millions of years of evolution, have adapted to this beautiful, fragile dot called Earth. Not just us, but everything else that's in here. When life goes from the pale blue dot and it just travels outside of it, it starts interacting with stressors and conditions that it just simply hasn't been adapted to. It's brand new, right? And there's so many of them. I'm gonna just point out two of them because they're some of the more evident ones here. One of them is the changes in gravity affect various biological processes in organisms. That coupled with increased exposure to radiation, okay? Those are two really important points because they essentially affect gene expression in organisms, which is really important. Spaceflight produces specific stress responses that allow Earth biology to cope with these novel living conditions. So what we're seeing is, even though Earth life hasn't evolved for space, we're seeing how Earth life is adapting to these different conditions. And it's really encouraging, it's really interesting, and it's also kind of weird sometimes. So moving on. So how does gravity here on Earth affects plants? Okay, so gravity plays a key role in plant development. And this is something that we're going to be able to see. And you, hopefully, if you're listening and you're watching, wherever you may be, might be able to experiment with this at home. Okay, plants use hormones that are affected by light and gravity to regulate the growth of not just the roots, but also the stems. Okay, so stems grow towards a light source, and in the absence of light, stems grow in the opposite direction of the gravity vector. All right. So, I mean, just got me thinking, it'd be so interesting if we have a plant that could grow in some sort of dome in microgravity and see all the different expressions there. But in the absence of gravity or in the absence of light, the stem of the plant is going to grow away from the gravity vector. Okay. That's how plants grow here on Earth. That's how gravity affects plant growth here. So how does microgravity affect plants? So microgravity affects root growth patterns as well as various other elements of plant biology. So since we're going to be talking about gravity mostly, so we're going to focus on that. But one way that gravity affects plants, gravity affects gas concentration gradients in plants. So because microgravity actually reduces convection in space, it causes spikes in gas buildup in plants. So this kind of goes into a set of feedback loops and then produces or restricts O2 supply, which can cause hypoxic zones that can affect rapidly respiring areas of plants, such as root tips. So the parts that are metabolizing, that are growing the fastest, could potentially be jeopardized by not being able to cycle through essential nutrients or gases because of microgravity. So this is important as we're looking forward, we're looking towards future experiments in microgravity regarding plant yields and genetic expression. Before I keep rambling on, Dr. Barker, you're way smarter than me on this topic. Is there a point that I'm missing here?
I think you covered all the parts rather well. So what's that diagram showing just there? Could you talk us through that?
Sure.
Just the picture itself, yeah.
Yeah, this was from Osaka City University, which is about 30 minutes away from here. So they were growing rice and Arabidopsis in microgravity. It wasn't too much of a focus on the root growth, but it was a focus on how the physiology of the plants changes. And it was interesting that in the microgravity, the Arabidopsis and the rice both kind of grew in just different patterns. They weren't as organized. Some of them seemed like from what I read, the stress in the microgravity killed some of the experiments that they ran. And this is just one of the images from the paper that I read from Osaka.
You have to send me the link to that paper or drop it in the link in the feed. I haven't seen this one before, but looking at it, it looks really fascinating. It looks like they've used like 60mm round Petri dishes. They're the same size ones that go into the brick hardware. Grown on an agar surface base. And they've actually got quite a high seed density. You can see it's almost like a lawn. You can, like the Arabidopsis, because they don't have any light, they're etiolating. So you can kind of see there's like a kind of silvery white streak through the middle of like a yellow top. So that's the hypercotyl elongating, and then the little yellowy like dots along the top, that's the cotyledons so that's the initial energy reserve that was in the seed that's now stretching up trying to find light, but it's not there. And on the ground, that's really kind of like, so I'm looking at C, the top right hand corner, that's really kind of like straight. But in spaceflight, you can see that elongation isn't really linear. It's kind of so that direction of elongation has been directed by gravity. That was like a negative gravitropic response that you can see on the ground, and that's really interesting. I've never seen that before, but it's totally predictable based on the gravitropism data that it has been produced on Earth. And that's the exact protocol that you'll be moving into, I guess. Yeah. So these data are really relevant to that. Like they would predict. So yeah, that's really cool.
Yeah, Dr. Hoson is a local, is a legend in Japan, and he's like one of the fathers of astrobotany in Japan. If I mean, the circles of astrobotany here are pretty small, but you mentioned that name and everyone just kind of shuts up. So he, I think he might be retiring soon. Anyway, brilliant guy. Almost every paper on astrobotany coming out of Japan has his name on it. So, but yeah. So that's how microgravity affects plants. Yeah, that's how it affects plants. So gravitropism is essentially plant gravity response. So both the shoots and the roots grow, or the direction that the roots and the shoots grow are a response to gravity. It isn't just the roots. Gravitropism is dependent on plant growth hormone called auxin. And specialized gravity kind of bead-like organelles called statoliths, which if you can imagine like and this is a demonstration that I saw Kai doing that I really like to do with my students all the time. This is just a petri dish. And can you see the beads there? Can you see me right now, or do you just see the camera? You see me?
Beads, yeah.
Great. So imagine this is a cell, and the little beads are actually azuki beans. These are these specialized organelles, and right now let's imagine this is one of the cells on this root. All of a sudden, the root or the plant gets shifted at a 90-degree angle. What starts happening is these organelles start moving and finding their way back down to that gravity vector, and that actually starts dictating how that hormone affects the growth of either the stem or the or actually that's particularly for the root. Stems has to do with something else, but basically high concentrations of auxin inhibit root cell elongation, so this causes the root to curve downward. So basically statoliths, they direct where the auxin is going to go, and then whether you're in the roots or you're in the stem, the concentration of auxin is going to affect the growth of the plant differently. So for example, the more auxin that you have on the root side, the less elongation you're gonna have. So what's gonna happen, it's gonna restrict that side, while the side that's got the least amount of auxin continues elongating, which causes that downward bend. And that's essentially how plants sense and respond to changes in gravity vectors at a very, very crash course simplified format. Basically, if you can think about it, wherever these little beads go, so will that hormone. Okay, so that's how plants essentially how they sense or respond to gravity. Now, which is the fun part, this is a simple gravitropism observation you can do at home. So I'm going to stop sharing really quick so I can see myself. Here we go. Okay. Hi, everyone. All right. So basically, this is something that we've run in the lab with our students here in Osaka. You can see gravitropism in action on Earth through a lot of different ways. There are devices called planostats. There's different stressor chambers that could be either complicated or expensive or just not available to people that are stuck at home. So I was digging around. I was trying to figure out what is the simplest, easiest, lowest cost method to try to demonstrate this to kids or to just have fun with it, because they make for really interesting photos. So all you're gonna need, if you can go on Amazon, I think you can get a pack of 10 for like $5. Get yourself a set of these square dishes, okay? You're gonna need a square dish.
There's a really good version of that. The chocolate brownies you can get from Target. They come in these plastic boxes that are the same size. You have to eat all the brownies first, but it's the same square shape. It's a little bit larger, but it will work perfectly.
It's fantastic.
It sounds like a challenge.
I had to do 3 reps, so I put on a bit of weight in isolation.
You get to eat a brownie. It's a win-win.
Now you got me thinking about brownies. Oh, gosh.
So, petri dish. Great
So either gorge yourself on brownies or go to Amazon, or you can just get a petri dish as long as you can mark where you started. Okay. So you got your dish. You're gonna want to get porous medical tape. Okay, some of that. If you have alcohol at home, rubbing alcohol, not scotch, some sort of ethanol, a seed type, I would suggest, just because I'm particular to this plant, Mizuna. They're a microgreen from Japan. They grow so fast, and the images that you can produce from this are fantastic. So if you can get Mizuna where you're at, order it. For me, that we've grown maybe about 20 different types of plants in this method, and the one that I've gotten most excited to see is the Mizuna. I know that Dr. Barker loves Arabidopsis, and it's fantastic, but I'm a little partial to Mizuna. Plus, I'm in Japan, so I got to put that push.
Mizuno is my favorite microgreen. You can and if you let it get established, it's so stress tolerant. Like, you can give it droughts and it will just turn slightly purple and become more nutritious. You can eat its flowers. Oh, it's a really awesome plant. I love it.
The first flowers that we had in our hydroponics in the lab here, it was just fun. The kids got blown away. But anyway, so what you're going to do is you're going to take your plate and put a thin layer of literally just seaweed based agar. Okay, no sugar, nothing, just agar jelly, nothing. Don't put anything on it. One thin layer of it, and then right as it's when it's almost finished hardening up, you're gonna want to get on the right down the middle axis of it and line up some seeds. Okay, line them up. Get a toothpick and clean a toothpick out with your ethanol. Oh, by the way, there's 2 ways of doing this. One, if you want to, properly sterilize everything as you should be doing, you can take your ethanol, take your seeds, do a quick wash in the ethanol, get rid of the ethanol, do a water wash, and repeat that a few times. You can do the same thing with your plate and then microwave it if it isn't sterile. We've done it at home, sterilizing it and not sterilizing it, and somehow it's worked out really well both times, and we haven't seen growth on our plates yet. I don't know why. It'll happen eventually, but that's why I said the ethanol. Anyway, you line your seeds, right? And for this demo, you're gonna want to have at least 2 plates. Have as many as you want, but at least 2. All right. Once you've lined up the plates, get your toothpick, slightly just push the seeds in, don't crush them, but just line them down the middle axis and press them down. Once they're there, you're gonna want to find a light source, like lean them up against a table. I'm gonna turn my camera here so you can see what I have here. This is actually a Arabidopsis for you, Dr. Barker. And you're gonna want to just put a light right here. We are. Put a light source and have them grow. Watch them every single day. Take photos. The most important thing is take your photos at the same time every day. Now, we saw that the plants they react to the changes in gravity vectors, right? So this has been maybe, this has been 1, 2, 3, 4, 5. This has been a week, okay? So this was our control. This one, we just let it grow. Let me see if I can get this so you can see it nice and without a glare. Let's see if we can, there we go. These are our Mizuna seeds. After about 6 days, this is what they look like. Their roots look really, really nice. They're growing downward. The stems are nice and healthy. Nice green, fantastic. Now, we took the experimental group, and on the 4th day, we turned it 90 degrees, okay? And this is what you would see. You see right there. All right, as you can see, the roots start finding their way back down. And what's really cool is you see the stems folding upwards, finding their way back to the light and actually reacting to the gravity as well. So this is this took less than a week. This is a really fun experiment that you can do at home. Just plates, lights, and time. Taking photos of it as you go along. We've done this at home maybe a dozen times since the quarantine started for my personal research, and because my girls here love it. I have two daughters. They're they're 6, and one of them is about to turn 4 in a few days, and they absolutely love doing this. The most important part of it was photographing it every step of the way. Now, this is here on Earth where that gravity vector is still there. We're just kind of, changing it, right? So what's important to us is what would happen to plants that are growing in a place that doesn't have that gravity. So let me go back to where I was.
Bill, what that reminded me of for some reason is like an Etch-a-Sketch. Old school, and for those that you know what an Etch-a-Sketch is, a little more slow moving, but could you make a little shape with your plant by rotating it around?
The plant would absolutely hate you, but I think it'd be really fun to try.
There's one extra thing that you could add to that. So after doing that the first time, perhaps like the students might want to change the protocol a little bit. What if you turned the plant on its side and put it in the fridge at the same time? If you put it down, if you cool it down in the fridge, it can't grow, it pauses its growth, but the statoliths will still sediment to the bottom. So it's getting a gravitropic stimuli, but it's not able to respond. If you then take it out of the fridge, like after a few hours, and then stand it up, you then watch what happens to the stems. You'll see something really interesting. Something just blow your mind. So this is a reason to practice this gravitropic study, like make it work in the way that Gil just described so well. And then once you've done that a few times and you've got some reps, you've got some confidence, there's always another way you can adapt it. And this fridge concept is one way. And anyhow, back over to you. That doing well, good.
Why didn't you not tell me about this? I would have done it already. That sounds fantastic. Guys, Richard, stop being so smart. Every time I talk to you, I learn something else.
I look forward to seeing the photos from that test, mate.
Let's see. So I think the next slide. Yep, these are some of the first ones that we did at home. And what's really interesting is that you're seeing in the bottom left, you're seeing that that the roots they're finding their way back down to the gravity vector, and you're seeing in the top left one that little bit of a bend. What was interesting, we've run this a few times, like I said, and that pattern in the middle one has happened about 5 times, which is a strange pattern. Don't really know what's going on there. Another very interesting thing that we've noticed is that, as you can see in the bottom one, the bottom row, there's always at least one that is really slow to germinate. They all germinate eventually. We haven't had a one that hasn't, but there's always one that germinates at a slower rate, which, is there a way that these plants are communicating? Is there something happening in that growth cycle that at that point they can sense the amount of nutrients, the amount of light, whatever it may be in the environment, and trigger or inhibit the growth of one of their fellow seedlings till the rest go up? I don't know. But it's
there is a really interesting research paper where they changed the density of seeds on a petri dish and actually able to show quite large genetic changes in the way the genomes are expressed based on the number of plants in the given environment, which would indicate plant-to-plant communication and a kind of quorum sensing, the community knows how big it is. If that then manifests in changes in germination rates, well, that would be quite a useful system because of the evolutionary pressures. If you've got an environment that's flexible, that's changing like it is in the natural world, it's often good to have some pioneers that poke their head up above the soil early, and some that kind of hang back and just check to see if it's safe, you know? So it makes sense both evolutionary, and the patterns hold true in the genetics. So what you're observing here is a very like natural phenomenon. It's fascinating.
Yeah, so this super simple, low cost demo that you can do at home, you can see gravitropism, you can see, plant growth. I mean, unless you really try to, you don't see plants growing like this on an everyday basis. It's really fun to be able to keep track of it. But there's so many other things going on there that you could take away from this little study, as long as you're documenting, photographing, and writing down everything that you see. It's been super fun to do with my students, and then that is a precursor to actual more serious layers of essentially the same kind of studies. So it's like you kind of tricking them into actually doing real science, because this is actual science, which is fun.
You can also then move them to soil and then grow them up and eat them.
There we go.
Experiment.
There we go. So this is something that Dr. Barker shared with me, and I want to share with you. So there have been a lot of studies on the ISS and on the shuttles and on various different missions on how plants grow in space and how gravity changes their development. So there is a device called the ABRS, the Advanced Biological Research System. It's basically used to run tests on organisms in microgravity in space. So plants have been tested and photographed and measured on this hardware multiple times. Richard, is there anything else that I'm missing on that you would like to add on this awesome piece of machinery?
Actually, it was only used once. It was used multiple times. Okay, on Earth, yes, but it just flew once on the space shuttle. But keep going, you're doing good.
I trust you. I saw a photograph of it on the ISS from 5 years ago, something.
I think it was broken by then. It didn't have a long shelf life. I think intergalactic cosmic rays flew for its computer, but
that looks like almost like a 2U or something like that. Standard volume.
It's very close to that. Yeah, I'm not sure.
I like the setup.
Keep going, Gil, and then I'll add some bits on. It's a cool setup there.
Cool. So for what it's worth, it did at least work once. And we have the images that came back from it here. So on the left, we're going to be seeing, let's go back to the video. On the left, we have the control here on Earth. And on the right, we will be seeing the growth patterns of Arabidopsis on the space shuttle. This is a very short video, so I'm going to play and then pause it if the internet decides to cooperate.
So this is the first time lapse of plants growing in space. The principal investigators were Rob Thurr and Annalisa Paul from Space Plants Florida.
What year?
2012. I'd have to go and check the databases. It was a while ago. Again, I think it was in the space shuttle era. The hardware then did go up to the space station, and Edison Blankenfall was meant to be the second person to use it, but due to technical difficulties, his experiment was moved into the Veggie system instead.
So I got the video working really quick before it shuts down on me again. So, yep, on the left is control. On the right, we have the one up in the shuttle.
You can see the camera reflecting there a little bit. So you've got a Arabidopsis seedlings growing there. You've got a couple of different varieties. There's one called there's one called Columbia, and that's from Columbia, Missouri. And on the right-hand side, on those two boxes, can you see those plants behave slightly different? They go straight down. So those ones over there, they're from the WS variety, which is from Poland, a place I can't pronounce. Worcestershire.
Yeah.
Sorry.
A lot of consonants.
Yeah. So you can see natural variation in the response to microgravity. One of them, the Columbia, you can see they're skewing. They're going off in a sort of different direction. But the WS don't. So you're seeing a different response. Sorry, keep going, Gil.
Oh, no. I mean, keep going. That's perfect. All that plus, I mean, the most obvious thing when you're looking at it is the patterns that we're seeing for the root growth, right? They're pretty different. What was interesting though is that both the control and the experimental plate, the roots were skewing to the left, and that was something that Richard pointed out. That I was like, "Whoa, that's super cool." They were actually. You can do probably a better job of explaining that I could. You wanna highlight on that point? It's pretty cool.
Sure. So well, Darwin was the first person to notice the skew as a response to both gravity and mechanical stimuli as plant roots go foraging for nutrients. Actually, there's a group in Italy that's been studying that to build robotic roots, and so people have been looking very closely at the foraging patterns that roots naturally form. So you can make roots go straight, you can make them go left, right, you can make them grow fast, you can make them grow slow. All you have to do is change their environment. And so they have a particular nutrient composition in their gel. They have a little bit of sugar, a little bit of MS, Magnesium scooge, mineral nutrients, and a very particular hardness. I think they used about 2% agar, which is relatively hard, so they use hard gel. And so all of these features combine to cause like a waving and skewing pattern. There's many different recipes that cause these different patterns. And so what we're seeing is a combination of environmental stimuli being perceived and computed all at once. The direction of gravity, the direction of light, the amount of nutrients, the gaseous composition of their surroundings. These roots are computing all of these different components and are then processing that and then changing their cellular elongation rates by changing their hormonal movement and hormonal interactions. And so this simple pattern in the roots actually tells us so much about what those plants are thinking and feeling. And I claim to one little extra clue. I've taken these varieties and I've grown them in very similar media and then lowered the atmospheric oxygen around them in custom-built low oxygen chambers and observed this increased skewing pattern. So there's many potential environmental factors on the space station and space shuttle that could have caused these roots to skew. Some follow-up terrestrial experiments have indicated that hypoxia could be that. But one thing is for sure, those plants don't have gravity. But their roots are growing in a certain orientation. The plant is growing in a certain orientation, which is very similar to that on Earth. So what's the next most important feature of the environment for helping plants coordinate their growth and development? What do you reckon?
Light.
Exactly that. And so this shows you one of the interesting, I don't know if artifact is the right word, but one of the interesting concepts with the ABERS hardware. The positioning of the lights next to the petri dish close up gave the plants a tropism that they could orientate themselves towards or away from. So gravity and light are just these two intrinsic features that have always been there since the beginning of the evolution of plants. And they've always somehow created this polarity between these two like North Stars, if you will. It's just fascinating.
Interesting, Dr. Barker. I've been thinking about an omnidirectional light configuration to completely confuse the plant and say, well, it's coming from everywhere. Now what are you going to do? The plants seem to be kind of smart, and if there isn't with the light, it's kind of leaning toward that if there isn't a gravity vector.
Well, this is why I got so excited by Diane's experiment with the plants taped to the window, because that's kind of what's happening there. It's that like light coming from all direction, kind of diffuse light environment. It's cool.
And all these different factors. I mean, there's the gravity part, there's light, there's all the different environmental stressors that are just in the play, and then we're adding the space environment. All of this is going to affect the eventual yield and the offspring of these plants if they were ever to have more of themselves in space. So that's super important. We have so much data on what happens to these plants. We don't know what it all means quite yet, and we certainly don't know what that means for future generations of plants and of space farmers. So this is why this whole concept of gravitropism, of gravitational studies in plants, is so important and integral to the future of astrobotany studies and likewise terrestrial agriculture. So just as a quick recap for your home experiment, if you're going to do this, write down your procedures, write down what you did, what you didn't do, what worked, what didn't work. Photograph everything. Just take photos of it. You can put it together on your phone, make a quick time lapse. It's fun. You can show it off to your friends over drinks. Look into those patterns though. Look into what's weird, okay? When you think of a plant, you have this particular image of what a root system might be. When, even if you're not used to looking at plants, if anything catches your eye as different, just write it down, then repeat. Write it down, rinse and repeat. And then after you're done doing that, do it again. Because these patterns start making themselves very, very clear if you're looking for them. Also, just the questions that I'm particularly interested in: what's affecting those germination rates, and how is gravity affecting your plants beyond just the directional growth? Because gravity is affecting all of your plants' growth, not just the direction in which it grows in. So that's what I would leave you with. At the most basic level, this is what you can do at home for however long we're in quarantine. You guys can maybe crack part of the puzzle as to how gravity fully changes the biology of plants here on Earth and in space. So with that, everyone, this will go. Let's grow plants in space. And my friend Kai, if you're seeing this, bro, I love your artwork. There's your spaceman again. Dr. Barker, thank you always for your awesome insight on everything. I don't know how to stop sharing. There it is. I'm done sharing. But yeah, gravity is important. Astrobotany is important. Guys, the last thing I'm going to say is we got to the moon because we learned how to farm. The agricultural revolution established humanity. And if it wasn't for that, we would not have cities. We would not have had rockets. We've gotten this far with the technology and the processes that we have, and we've kind of depleted our stock, or we're heading that way. The future of agriculture will determine the future of humanity. So this research literally pays into what we're going to be doing as a species for generations to come. So get curious, grow plants in space, eat your veggies, and stay healthy, everyone.
Awesome, Gil. Thank you so much. Big shout out from Japan there, giving us a good background on some simple experiment you can do at home. And, you know, some people might even have difficulty getting some of these basic materials. So I'm wondering if anyone, I know I've got a number of teachers here with us today. I see Sam and Lori have jumped in since I last looked. So good to see you guys. How might we use the study of the gravity as we kind of rotate these plants around, and maybe not, what if we didn't have these plates available? What else might we do? Can we MacGyver something? What do you guys think? Yes, Diane.
So that's one of the things that I've been investing a lot of time in, and collecting all sorts of different things that people and teachers can use, because we don't always have the money in the classroom. So with that in consideration, I started putting together literally 2 boxes of materials for here and then for later when we get back to work. So some of the dishes that are really useful are when you order food, and if it is the type of dish, maybe I should grab some of those so you can see them. that is dark all the way through for the later process when you're putting them in sort of like a ground effect so that it's dark on the bottom. Those dishes are really, really great. Do you want me to grab one or two of these items so you can see them?
Sure. Anyone else want to weigh in while she's doing that? As far as this is amazing, great place to start and think about a simple experiment.
I've got one. So again, I was really excited by the plastic baggies. Don't get the ones that have the pictures of superstars on them or like Avengers on them. You want to get the perfect clear like seal up baggies. They come already sterile. The next thing you want is some kind of growth medium that's gonna hold itself together. I tried it with like vermiculite, but that will like sediments to the bottom. So just like filter paper works, but be careful of white filter paper because it's been bleached. So I've lost a whole semester of my life. So you're using this white filter paper and just watch plants die after die after die. And I kept thinking I was doing something else wrong. But the process of making paper white is a bleaching process, and bleach is what we use to sterilize seeds, and it kills them if you leave plants in them for too long. But those plastic baggies like that Dan was showing the other day are brilliant, and I really recommend trying them with filter paper inside them. But not the white bleach stuff because that will kill. But
hey, Lori, you had an idea here about using some other types of paper. You want to jump in?
Well, the traditional elementary level is you just put a damp paper towel in the Ziploc bag. So he just answered my question about the Ziploc bag is already sterile, but maybe not the white commercial paper towels, but the brown ones from school or the brown generic coffee filters with the coffee filters.
Oh, coffee filters. I got some of those.
So that might be a possibility.
Lori, cotton balls work great too. You've probably done that already, where a lot of the teachers will put cotton balls inside the plastic bag and then put the plants in. That's a really good one. Right now I have a director of a program who is teaching food, and she is teaching all about the science of food. And one of the things she's done is she's created with newspaper, simply newspaper, nothing else, a folded origami cup. And they're growing in the origami cup. So she's going to be sending me that. The directions for that, because that would be really, really interesting to see. And I would maybe line that inside, depending on what her results are. So the plastic Ziploc bags come in a million different shapes. And the next ones are going to be really large for me because I'm going to be dumping in gravel and all sorts of stuff, including, the Mars simulant and moon simulant. So there's a lot of cool things to go with. I think, Richard Barker, you were mentioning something like that. That's so these are actually berries. They weren't brownies, sadly. The other thing that I use from food containers are these very dark containers, and they're heavy. They're very resilient. They're hard. Traditional in school, especially in the kindergarten classroom. Egg cartons. All right. This one is 2 dozen, so you can put a lot of plants in that. Styrofoam, little tricky because we don't want anything leaching into it, but if you use the recycled container and you line it, all right. The one thing that we do have to do with this though is punch some holes in it because water has to drain through, and the same thing with that container. Simple starter, pill boxes. So when you have your medicines come in, great place, the cap's already there, and then away you go. And another one is there's a whole lot more, but this is what I'll show you for today. That's the double garden that I talked about the other day. So right now on the bottom, one of them has gravel, the other side has eggshell, and this is what we buy prepared lettuce in. So how do we support that? You get another box and cut off the top. I'm sure you have your holes in there, and maybe something to sit it on so it can drain into, and a cookie pan, and it sits on the cookie pan. So there are a million different ways, and that's a whole other thing to keep looking at. I will be creating a lesson plan for that for Magnitude, so you can look forward to that.
I love it. That's great. And Lori.
I wanted to say that one thing that is really big in our community, since there's so many people that recycle, is using the cardboard. And I'm really intrigued by the newspaper origami, because then you can take it and directly plant it without having to transplant it. You just put the whole container in the dirt, and the cardboard or the paper just becomes part of the soil composite in your garden. And so it's really easy for kids to transfer. I've been using the peat pots and then put them in a plastic cup, and then just take the peat pot out and do it. But I think the paper would break down easier. The peat pot tends to cause things to be a little bit root bound when kids want to transfer them.
Awesome.
That
Thank
would
you.
be the same thing with the tea bags, Ted. So the
Mhm.
tea bags, although they're in the plastic bags now and growing the plants, we would transfer them directly into the earth while they're sitting inside the tea bag. It's completely compostable.
Well, I've got a great segue for us here. We're talking about how we can put different containers to run our experiments here on Earth and think about gravitropism. We're joined, who just logged in with us right now, is David Kasuma from Tupperware. And they're actually solving this problem in space on the veggie with something called the pond system. I think David can probably share it in a much greater way with us. David, we've got teachers from around the world with us: Japan, Germany, Canada, representative for our Central and South American countries, South Africa here, and some teachers, of course, across the U.S. Dr. Barker from Gilroy Lab as well with us as well. So, David, thank you so much for joining us. Would love a little background on maybe on what you do there and how Tupperware got ending up doing stuff in space and what your current project is about. We're really pretty excited to hear from you.
Okay, thanks, Ted. Can you hear me?
Yep.
Okay, perfect. So greetings, everyone. I'm really pleased to be here to make a presentation and to support this initiative by Magnitude. You know, it's one of the things that Tupperware always feels is important, is to support the education community. You know, and certainly this is an opportunity to support with a theme that's really, really motivational, you know, and really encouraging, especially for young people. I lead R&D at Tupperware. So my job is to come up with new materials and also new technologies as we drive forward to products of the future. We had been working with NASA, actually looking at some of their tech transfer opportunities, because NASA develops a lot of technology. And they had invited us to Kennedy Space Center about 3 years ago, where we started to look through their portfolio of offerings. And one of the things they asked us was, you know, they had this project called Ponds that was being developed. It was actually an internal competition between NASA scientists to try to develop the next generation growing system for the International Space Station. And they came and asked us whether we would be interested to work with them. You know, of course, it took us a little bit of time to think about it, because it's not something that we do every day. We don't normally get into space programs. You know, our interest is more to utilize technologies from space and bring them back down to Earth. But it was an opportunity to do something different, you know, so I was able to convince our senior management to go in this direction, you know, and then they put us in touch with a company called TechShot, who is the, I don't know how to, their implementation partner, who basically prepares everything for space flight. And then we had been designated as the equipment supplier for this project. Yeah, the rest is history, and I have my presentation here. Should I share that, Ted?
Yeah.
Okay, let me do that. Let me share my screen. Oh, I'm actually too far. I was just reviewing these slides before I came on. Okay, can everyone see my screen?
Yep.
Okay, good. So this presentation is for Magnitude Live, developed specifically for this group over here, and I hope it tells you a good story. So I work for Tupperware Brands. I've been there for about 18 years, leading R&D. But I'm also a member of the World Design Organization. So this is a world organization that connects all of the different design disciplines around the world, from corporate designers to association designers, people who own their own design firms, and even universities. And we focus on the UN Sustainable Development Goals. I'm sure most of you know about them, to focus on how we can develop products that have meaning and that have impact in terms of designing for a better world. So at Tupperware, we focus on everyday good design. And this is just to give you a few slides of historical perspective in terms of what we do and why we do them. We develop a lot of products. We have 3 development centers. I guess we call them design centers. One is at our headquarters in Florida. The second is in our European office in Belgium. And then the third one is based out in Singapore. Our focus is on everyday good design. And what that means is that we are not so interested in developing products that end up sitting in museums. A lot of designers like to have these extremely beautiful products. We do as well, but what's more important for us is that the products are very useful. They're good for every day. And our real interest is to come up with a product that is used every day and multiple times per day. And this is really important because the more people use our product, the more impactful we are on their lives and the more we can improve their quality of life. All of our products are sold with a lifetime warranty, and this is really important for us as well because a lot of people know that we don't play at all in the limited life products that are sold, in this category. Everything that you purchase from Tupperware, we like to imagine that they last a lifetime. If you are looking for a definition of lifetime, it's at least 50 years of continuous use, meaning that if it's designed to be used every day, you can use it every day for 50 years. So my screen seems to be a little bit stuck. Hold on.
And queue up those questions too. If you're on YouTube, feel free to ask a question in chat. And if you're here, feel free to use the chat inside the Zoom as well. And we'll bring some questions in. If you want to end your presentation and restart it again, you can do that.
So let me see. I'm somehow connected through Zoom, but my keyboard is frozen. Hold on just a second. Sorry about that.
That's quite all right. Maybe it's the windows. You know what I might be able to do is, or I think Tony has the comm.
Oh, here. We're good. Do you see the next slide?
Well, you had to get to space. That was the problem.
I had to get to space, and sometimes it takes a little bit longer to get there.
Yeah. Past max Q, you're good to go.
So our journey into space actually started in 2005, quite a number of years before we started working with NASA on the PONS project. Here at this time, we were connected or contacted by the ESA, the European Space Agency, who came to us. This was 2005 was during the year that NASA was not flying because of the Columbia disaster. But they had a project that was a developmental project from a university in Italy to develop stem cell research that they wanted to try out in zero gravity. So they asked us, would we like to work on this project because they needed a container that had very specific dimensions, and they wanted to know whether we were willing to build it for them. But Tupperware has been around for quite a number of decades, so I have access to our global mold master is what we call it. And we have over 10,000 injection molds in that database. So I was actually able to find a container that was off by, I think, 1 millimeter on one of the 3 dimensions that they were looking for. So it was really close. I proposed it and they accepted it. You know, we had to do a number of tests with pressure, with vibration, and also to make sure that you know, there was no off-gassing if the unit were to burn. Finally, the container was approved for space flight. A lot of paperwork during that time. This was our first exposure and we were just really overwhelmed. That's why when we started to work with NASA and having the opportunity to work with a company like TechShot, we actually didn't have to worry about any of this paperwork because they took care of it for us. So, you know, Tupperware normally is not developed and is not designed to withstand, you know, such critical pressures or vibrations. But they were allowing us to modify our containers so that we could customize them specifically for this launch, which is what we did. You can see some of our prototypes with these bicycle valves, just testing pressures. You know, we had to test everything internally and then externally in order to get everything approved. And then if we skip forward, this was Commando Roberto Vittori, because it was led by an Italian astronaut and taking out the Tupperware container on the International Space Station. And this is back in 2005. It was interesting because for me, it was the first time we were connected to spaceflight communications. Every time the Tupperware container got moved, I received an email. You know, I got an email that said, the Tupperware container has been taken off of the TMA-6, which is the Russian spacecraft, and moved into the International Space Station and has been implemented into the Aquarius incubator. You know, you can't get a more cool email than that one. A lot of publicity came from that one, but most of it was in Italian because of the Italian space flight. So it was interesting for us. We just documented some of them. I want to talk just a little bit about why NASA was interested in Tupperware on this project, you know, and that is because a lot of our products are designed specifically for food conservation. So they like the aspect that we've been developing products on utilizing food safe materials for decades, because for them, they wanted a product that was food safe. The second thing is, you know, we've been focused on food conservation, which means that because of some scientific studies that we've been doing, mostly with universities that are focused on post-harvest food storage, you know, we can actually provide longer life to food, or let's say longer freshness to food. And it's really important. If you take a look at this slide, it shows one of the real pain points. You know, 40% of food in the US ends up in landfills, which is a huge amount. And if you look worldwide, 1.3 billion tons of food gets thrown away, you know, and if you imagine all the people who don't have food and who are starving, it's a huge number. So we take our motivation from real life situations. And we have this product that we developed, you know, and you see that it's categorized into 3 different columns. Because based on these tests that we had done with land-grant universities in the US, they were able to categorize the vegetables and the fruits into high breathers, into low breathers, and into somewhere in the middle, medium breathers, which meant that if we took the respiration rates of these vegetables into consideration, you actually get a net positive benefit in terms of being able to keep them fresher for a longer period of time. Everything when we first launched this was in words, and then we switched to icons because we sell in over 100 countries around the world, so it's difficult to translate everything all the time. And here you can see that you know, when people put their fruits or vegetables inside these containers, they can customize it to their own needs. You know, so the little slider on the top basically hides holes that are on the inside. You know, and depending on where you slide it, you'll either get it totally shut off, or you'll have it open up to the maximum amount or somewhere in the middle, which is half opened. It's interesting how everything with regards to science on this project boiled down to the size of a hole, size of an opening versus the volume that you were trying to store your food. So this is just a test showing 21 days, and you can see the difference between what's stored on the inside and what's stored on the outside. We've also, sorry, go ahead.
Oh yeah,
Ted?
David, I think Javier had a question about respiration or photosynthesis. Javier, do you have a question?
Yes, so according to you, you have different rates of respirators. So are they doing photosynthesis after the vegetables are cut and fruits?
Yeah, so this is the part that a lot of people don't realize. When they go to the supermarket, they think that all food should be stored the same way, but it's not true. The fact is that the food, especially the vegetables, are still alive, even when you purchase them in the supermarket. The length of time that they actually last depends also on when they were delivered to the supermarket. So really, that's the only unknown. They could have been delivered yesterday, or they could have been delivered 2 weeks before. So that'll play a role as well. But because they're alive, they still will continue to respirate. Okay. So we've also done a number of work with cheeses. This is looking at the high moisture cheeses in Europe. And what we found was by working with a company that made special membranes out of plastics, we could design in a certain transmission rate where we moved heavy moisture on the inside because the cheese was evaporating to the outside. Otherwise, you end up getting a drip back on the cheeses, which is not so appetizing. So those of you who have been traveling, through Europe know about these types of cheeses. The Europeans will tell us that in the US, in America, we don't have real cheese. Only they have the real cheese. But this one was very good because it uses a very simple physics principle. You basically move the moisture from an area of high vapor pressure to an area of low vapor pressure, and it's always the highest on the inside because that's where evaporation is taking place. And then we also worked on conservation for breads, which was a very, very difficult project because the product just needed to work. But you know that if you go to different countries, in Germany, for example, the bread tends to be very high in density. So they always have a harder bread than most places. And then in France, for example, just across the border, they have soft bread on the inside with maybe a nice harder shell on the outside, but it doesn't really matter.
Great, thank you.
Okay, sure. And then now I want to come quickly to this project with NASA and TechShot, which is called PONS. PONS stands for Passive Orbital Nutrient Delivery System. And when we first started on this, we wanted to make sure that it was space friendly, of course. And again, this whole concept was to develop something that would be second generation. NASA, as you know, has been growing plants on the International Space Station now for quite a number of years. But there are certain types of vegetables that they haven't had good results. For example, tomatoes. Tomatoes is one of the types of vegetables that consumes a lot of water. And the current pillows that are up there have not been able to get good results. So, this is one of the reasons they were looking for something that was a little bit more flexible, more adaptable, and also with the input of crew, they could achieve different types of food that than they were able to achieve in the past. One of the things you should see here on the bottom, Tupperware interest. We have also an interest to take this technology eventually and create green kitchens of the future. This is also really important because green kitchens is very on trend. A lot more people like to have immediately at their disposal their herbs and and various vegetables that they can use in their cooking experience. This is the roles and responsibilities that were finally developed. So TechShot was selected by NASA as the implementation partner. So they are the main contractor, so to speak. Tupperware was designated as the hardware developer. So all of the hardware for this project is produced and manufactured by Tupperware. And then, of course, we had to connect with KSC and also Johnson Space Center as well for the program management. When we first started this project, we really didn't know what we were getting into, but we got kind of blown away because NASA really opened up their doors and they gave us access to a lot of places that we didn't think we would have access to. So this is in their life sciences laboratory at KSC. And we were able to go and monitor almost any time that we wanted to. We would have to make an appointment, of course, to see the ground level testing that they do. So normally when they send something up onto station and they want to do a control, they would be replicating the experiment on Earth as well, normally in the life sciences lab. So each of these large environmental chambers that you see in the back are rooms basically that you can step in that has fully controlled environmental conditions. It's almost like the Exo Lab, just in life size.
Wow, that's pretty awesome.
The fellow that you see wearing pink, so that's Dr. Levine. He's the original inventor of the ponds. So you can just see some of the photos from our first visit. This is the cargo. I think it's called the bay, some cargo bay where they
high bay.
High bay. That's it.
High bay.
Yeah, where they do a lot of the assembly. And this is their cryogenics laboratory. You can see some of the Tupperware products that are on the table, and we were just testing them in the extreme conditions of temperature. How high can we go? How low can we go? And see, just at a very base level where our starting point was. If you want to read a little bit more about our project, you can also go to the NASA website, or you can just Google both Tupperware and NASA on the same line, and it'll take you to our project. And it'll explain to you in detail, about how what they claim that Tupperware wants to turn astronauts into space farmers so that they could grow fruits and vegetables that they can consume in space.
Yeah, space farmers.
Yeah. So this is the original prototype that Howard Levine was working on. So you can see that it's a collection of items that you can find in a regular hardware store. So most of these things are PVC pipes, for example, that were cut and manipulated right in a fabrication laboratory just to get the basic functionality done. None of this, by the way, is approved for food contact because we would never use PVC for food. But you can see on the right-hand side, there's basically what they call a root cylinder. And then on the left-hand side is the reservoir that everything fits together on. And these were some of the early ground tests that were conducted. You can see tomatoes on the left-hand side. So they were quite pleased that tomatoes came out quite well. And then all sorts of lettuce was tested as well in this preliminary unit. Of course, you all know about the veggie, and the ponds was designed specifically to fit within the veggie. Basically, 2 rows of 3 for a total of 6 units per veggie storage. And we learned a lot along the way as well. These were some of our early prototypes, and we had to do a bunch of tests with regards to things like irradiance to see, the reflection of the light and whether the plants at a certain distance would be collecting enough light for its growing needs. And what we learned, as you can see over here, is that NASA had some very specific definitions. They were looking for materials that were fully opaque. For Tupperware, opaque just means that you can't see through the material. But then when you put it through here, you could actually see some of the lights through them. And, NASA was saying, no, this is not opaque, because to them, opaque means a 100% blockage of any light transmission, right? Because for them, this was very important because they wanted to make sure that any water that was stored in the unit did not start to grow mold. Apparently mold on the International Space Station, where you have a closed environment, is something that's terrible, that's very bad. So this is what the unit ended up looking like. You have the reservoir on the left-hand side, and you have the root cylinder on the right-hand side. We started calling this a cupcake because it kind of looks like a cupcake. On the top, it has the germination cap so that when you are just germinating a seed, you can keep the environment with much higher humidity. And then this is basically the breakdown. I think we took almost, I'm trying to think, some 40 or 50 parts. And then we consolidated it into 5 main parts. So you can see that there's the base, there's the reservoir, there's the bottom of the root cylinder, there's the collar, and then there's the germination cap. All the other parts that are in there are basically purchased and then added to the unit afterwards. And then this is the breakdown by system level. This was actually prepared because everything was required to have a part number, and everything is assembled in a way that the astronauts would have no trouble finding out which piece was what, and how to operate the unit. So this shows how it would be placed inside the veggie, 2 rows of 3. Just side by side, each one holding only one plant. So here we just pulled out some samples out of the test chamber just to show and take some photos along the way. This was from a recent ground test that NASA conducted before launch, and this was really a good indication in terms of how it would perform in space, because the only difference was the gravity. One of the things that we found out in working with NASA as well is that they gave us access to their various research centers. And one was the Glenn Research Center that has most of their knowledge in fluid dynamics. And we had one of the people who are now, I think, Mark Weislegal, who is leading the University of Oregon, I think, University of Oregon in Portland. And he actually had built a small version of a zero gravity chamber that could test some of these things, almost mimicking a demo flight, one of those parabolic flights where you can see how things react in zero gravity. So, we were able to develop this with specialized geometry, with surface energy, and with capillary forces in order to get the water where the plant needed it. Because on Earth, it's always easy. You have a gravity effect, which means when you pour the water into a potted plant, the water goes all the way down to the bottom, the roots go down and the plant goes up. but in space, the water wants to go everywhere. So that, that was the big engineering challenge. How do you get the water only where it needs to be so that the plant can continue to draw from it and can utilize the water in real time, meaning only as it needs it. So this right here shows some of the assembly prior to space flight. So you can see that some of them have the white shrouds. The white shrouds actually cover a transparent piece, because even though the units are designed to be fully opaque, in the cases where you are doing initial testing, NASA wanted to see what was actually happening in the root cylinders. So that's why we have these white shrouds, so that it can be opaque, but we can open them up as well and see what's happening on the inside. Okay, so this is actually from the first launch, and we've had since then some on-console meetings with NASA as well, where we were able to go to the Kennedy Space Center as they were talking directly to the astronauts working on our project. So that was quite interesting. We weren't allowed to speak with them, but we were allowed to submit questions that they would ask on our behalf. So this is actually Christina Koch, who recently came back to Earth, showing a photo of when she was working on the ponds on the International Space Station. And then this is just one final closing thing. Water is also a big thing that we focus on. And, we have our resident astronaut here, Story Musgrave, who is best known for doing the spacewalk to fix the Hubble telescope. So he's actually resident here just south of Orlando in a town called Kissimmee. And he's worked at Tupperware for about 5 years in the past, focused on water, and here is looking at developing a personal water purification system. You can see that this is in March 2009. If we fast forward to today, a lot of people don't know, but this water product that is focused on cleaning water, and it does a really incredible job. It has a 6-log reduction of bacteria, and it has a 4-log reduction of viruses. So 4-log is 99.99% virus free. 6-log is 99.9999% bacteria free. So it's not an inexpensive system, but it cleans water like no other unit does. And a lot of people don't know this, but if you were to ask me today, which is our #1 product in terms of sales and volume, it's actually water. Okay, I think that's the end of the presentation.
Wonderful, David. Thank you so much. A question about that last product you showed. How many gallons of water or liters of water can go through one of those, and what kind of filtration systems actually used? Usually that gets clogged up after a while, right?
So this one is designed so that the filter medium, so the component on the inside that needs to be changed, we call it the consumable, it only has to be changed once a year. And it actually uses the NASA technology. It uses a nanoceram wrap, which is basically a membrane that's made out of ceramic material. And the way that it works is that it has nano-sized pores. So it actually removes pathogenic activity mechanically. So that's why it works really well in the field. Because people realize that you don't actually need to use chemicals to kill pathogens. You can actually extract them mechanically.
And no power required for this. Is just, since it's mechanical, you're just flowing water through it. Is that right?
So no power required to clean the water, but we do have a battery in here. And the reason why is because we have a flow meter. We want to be able to monitor how much water actually goes there, so that people know when they have to change the cartridge.
Oh, got it.
So there is a flow meter that manages that, but otherwise it's it uses the power of the pressure that comes from the tap.
The fun adage on the station: yesterday's coffee or today's coffee is tomorrow's coffee.
That's right.
You technically can't use that in the same way here, I imagine. There's other processes going on.
Yeah, I think it still is a consumable, so it still requires you to change it. The interesting thing about this one, Ted, is that when you have a filter that is high performing, it's non-discriminate, which means it takes out the bad stuff, but it also takes out the good stuff. So actually, the real benefit of this unit here is that it also has another chamber on the back end, which means on the outflow, that goes through a series of what is called Bahukan rocks. It comes from Japan, and it's the only substance, it's the only mineral substance that's actually been approved by the Japanese government to add minerals back into water, because you've now taken out the good stuff, which is normally the minerals that most people want in the water. This chamber actually puts it back in the water again before the outflow, so that when people drink the water, it's mineral rich again.
Well, that's super cool.
Wow.
Yeah.
Hey, so can you unshare your screen there?
Oh,
And then
sure.
let's open it up to our guests here in Zoom. And also I'm going to check YouTube as well, but floor is open, guys. So you got some questions for David about space or about Tupperware? Javier, go ahead.
Okay, so you develop technology and methodologies to grow and develop these ponds units. So what are the components that you're going to be able to put back on Earth and develop a product that is probably consumable for the mainstream people?
Sure. First of all, the ponds is designed in a way that every component is reusable, which is not normal for NASA, because NASA normally will send something up, use it once, and then discard it. But everything here is reusable. It's up to NASA whether they want to reuse it or not. But the interesting thing for us is, there's an opportunity because this whole ponds unit is designed in a way to make it super efficient. You only have a small growing space. In fact, the reservoir is only 500 milliliters of water. So it only lasts a couple days with regards to the plant that is consuming the water. And then the astronaut will come and meter in a certain amount, specifically every few days in order to top it off. And what has been done in the life sciences laboratory on ground tests was replicating exactly what would be done up in space. So we've taken all of that learning, and we have the opportunity now to utilize it to build a green system on Earth. We just thought it would be interesting to offer consumers the opportunity to buy something that would be similar to what's up there, and also optimized in the same way, except that on Earth, you still have the gravity effect, which actually means that it should actually perform even better on Earth, just in the orientation that plants like to grow, because plants normally grow in natural order. We haven't actually come yet to the point where we have designed a product. It's something that we plan to do, let's say, within the next couple of years. But for now, we have made the agreement with NASA that we'll focus only on their project and help them get this equipment approved so that they can use it for deep space.
All right, thank you.
Hello. Hi. So I'm an old friend of Howard Levine's. I was wondering, are you looking at different intercropping systems, or are you just have you been testing different varieties of plants in there and different combinations of them?
So the answer is yes. The main components that I showed, which are all of the housing components, the external component and also the internal components like the root cylinder, those are all the same. However, we've been working together with TechShot and with NASA to change the configuration of what goes on the inside, because inside the root cylinder is actually a combination of capillary mats, which is It's like a cloth substance that is very efficient at wicking water, and also a component called arsenite. It's almost like a clay-like substance, but it's heavily fortified as well with nutrients that plants would need to grow. I should explain a little bit that we have launched what we called 1.0 in the very first launch. Then there was 2.0, and then recently in February, we launched 3.0. And the difference is that when we launched the first one, it was after a very extensive ground test that showed 100% germination, so that was good. But when they actually tested it on station, they found that they had 100% germination, but the seeds were overwatered, according to them. So they came back and they asked us, "Hey, can you do a little bit of work in order to restrict the water?" Maybe, the way that they described it, and I think they were being kind, was that you guys did such a good job at transporting the water that we overwatered. But so the next one, we ended up restricting it because we created additional layers, which were using some polymers that created some barrier effects because they were also hydrophobic. And then we had the shutdown of the government in the second year. So unfortunately, because of the shutdown in the government, we were not able to fully test version 2.0. And that flew. They made the decision to still go ahead and fly it. But that flew with very minimal amount of testing. And what they found out when it was up there was that it was underwatered. So it's interesting because TechShot always likes to describe this project as almost like a case of Goldilocks. First it's too much, and then there's too little, and then can you redevelop it in terms of the insides so that we can just meter in what we need. So version 3.0 was basically trying to find a middle ground between those two that would help the plants not be over or underwatered. And there were 3 different versions, just to answer your question, that were flown. NASA only wanted to fly 12 units. 6 of those units were the version that Tupperware and TechShot worked on. And then 2 of the other versions, which is basically 3 of each were flown as well to make up the full set of 12, were from NASA. So those were some of the ideas that Howard Levine had that he wanted to test out. So that's currently what's on station today.
A little competition going on there, huh?
Yeah, a little bit.
Yeah, well, that's how you end up with a better product, right?
Yeah, so the main components don't change, but the internal substances do change, depending on what you're trying to do.
Cool. Now, there's a question that came, or comments from both Sam McCoy and Shivar. You want to ask the question, Shivar or Sam, with respect to use of these devices, maybe in the classrooms themselves? So is the PONS device available to the public or to researchers beyond the NASA work that you're doing?
I think we would have to ask NASA for permission. But I mean, as far as Tupperware is concerned, if NASA says it's okay, then for us, it's probably okay. But just to give you some framework, cases had come and asked whether they could utilize some of these PONS units for some of their work on the National Laboratory. And my understanding is that NASA gave them the okay. So, I think once it is standard equipment, it'll probably open up. I'm guessing.
Well, there you go. Shivar, you want to?
Yeah, exactly. No, because I was saying, how can we take something like this and use it as an educational tool as well? Because the pond system is looking at water, oxygen distribution, as well as root growth. So it's perfect for all of those things as well. So how can we take something like that beyond just NASA and implement it into the classroom or something like that for students?
No, we would love it, but again, NASA holds the keys to that because the whole project was done on their behalf.
So I have an idea, guys. If we want to, maybe proactively, we can get together here as a global group and make a request to NASA that we would like to experiment with this type of technology. Now, it may go nowhere at all, but also a door might open. So it sounds like if NASA says okay, then we have to figure out what the costs are and other things like that. But wouldn't that be cool?
Yeah, the other thing we can do as well, Ted, is something that you and I discussed, which is maybe sometime in the future to develop a version of ponds that fits in the ExoLab.
Yes, yes, yes.
Yes.
You know how hungry we are for that.
So right now, this current one is too large for the ExoLab because it's been designed specifically for the veggie. But if we put our heads together, I'm sure we could come up with a good solution.
Well, didn't we have 7,000 different models or something in your database? I'm sure we can find a.
Yeah, over 10,000.
How many?
Containers. Over 10,000 containers.
10,000. Okay, there you go.
Hey, David, I just saw a lot of head nodding.
Yeah. I'd also like to throw my hat into the ring here too for testing those. So I work at UW-Madison, so we're friends with Joya and all the folk at KSC. We've got things going up on the next couple of flights, and the future of space exploration is growing vegetables. The pond system looks like it's like that next level technology, and so again, we would love to, as active astrobotany researchers, we would like to be able to test the hardware to see if we can use it in future like NASA grant proposals. Because you kind of need the preliminary data before you can put in a proposal to say that you have a insightful experiment that you'd like to use in a particular piece of hardware. Most people that put in research grants go in on the cautious end of the spectrum using flight certified hardware because you know it's going to go through the peer review process. And so having access to a pond system would allow us to develop these preliminary data for future proposals.
Yeah, that would be excellent because Joya Mas has also been very involved in this project as well.
She's awesome, right? So I'll reach out and see what we can, and because we can speak to the people at KSC as well, and get that ball rolling for the educational stage of this program to get that kind of permissions that the rest of the community here were asking for. So like working down through the researchers to the teachers and to the students and trying to get that ball rolling. If that seems
yeah, that'd be great. And both Howard Levine and Joya Mas would carry a lot of influence, I'm sure.
And David, just for context, Dr. Barker is our patron saint here that supports our beginning understandings of what might be possible as we think about astrobotany, whether it's for growing gardens on Mars or doing something on the space station, or probably most importantly, providing food for 8 billion inhabitants here on Earth. So yeah, anything that we can collaborate with at that scale with research from with Dr. Barker to our kindergarten students or high school students or college students, like I see Sarah from the University of Florida is here with us. And it's always exciting to see all these different thoughts and approaches to solving these various needs.
So University of Florida was one of the land-grant universities that we worked with as well to develop the food conservation containers. We worked with your agricultural team in post-harvest technology.
That's very cool.
Yeah.
It looks like Diana has some comments.
Oh, yes. Diana, go ahead.
Go ahead, Diana.
Actually, more of a question. I did put a link in of David St. Jacques, our Canadian astronaut who did work with the ponds on the ISS, along with the American astronauts. But I'm curious to know, he was filling the reservoir in that picture. So is there something in the device that actually tells the astronaut it needs to have the water filled now, or is it the judgment of the temperature, the light, the moisture? Is it similar to what we do on Earth? So how does the I guess, is there a communication between the astronaut and the ponds unit, or is it something completely separate so that they know when to fill the reservoir?
So the ponds unit is designed to be passive, so it's not an active system. It doesn't really talk to you. It is designed not to use any additional energy than that's already required in the veggie system that creates, the energy from the LED lights. But the way that it works is that through testing, they've determined how much water needs to be metered into the container after a certain amount of elapsed time. So I think that things may be adjusted when the life sciences lab or some a group at Kennedy talks directly to the International Space Station, specifically working on the project. It's possible they might make some modifications at that time, but otherwise, it's more. How do I describe it? It's like a scripted activity.
I had an idea regarding this though, because we went through this with the Zinnia Grow Out with Scott Kelly, where you're phoning up and like Joy's getting out of bed, running down, going, oh no, it's flooded, and the astronaut giving up his time to like work out just how to tweak the protocol for the alteration of microgravity. But what I was thinking when I was looking at the pond system, you could kind of have a clear like plastic cylinder on one side and fill it with those crystals that expand and shrink when they get water in them. And you could imagine using that as like a proxy for like how to measure how moist it is inside. Because the system itself looks like the astronauts can't see inside because it's all white, because you don't want loads of like algae and stuff like that to go inside. So it's got to be like light tight. But you could imagine like just having a slither on one side where you have this cube. And I've seen at Home Depot these crystals that expand and shrink to hold more water or less. And if they had like a porous membrane, so they could get the same moisture content that was in the arsolite, you could imagine them expanding and shrinking, showing that like clear cylinder being more full or less full of crystal. And so you'd have like a proxy for how much like water is inside. It could be like a gauge.
It's an interesting idea, Richard. I think definitely it could be an opportunity to do some testing. It's not something that they've looked at, I'm pretty sure. I'm not even, I think the first designation would be to find out whether those crystals are food safe, are food compliant. But otherwise, it sounds like it's something definitely interesting to test out.
Jolly good. I love designing experiments, so.
Yeah, I try to find a source on that, and I just saw industrial applications, but no place where I could buy like a kilogram of it or whatever.
Are you talking about the crystals, Ted?
Right. Yeah.
Okay, so I have a number of bags of them here because for some reason, in my crazy science days, I bought a whole bunch of packages that go with the kit, the teaching tank. So I can certainly package some of that up and send it out to you.
I guess I'd want to understand the materials and composition, and to David's point, food safe, right? What
Right.
leaching, whatever.
It comes with a descriptor inside each bag, so I can provide you with that, but I think a little more investigation into it might be worth it. But certainly they do expand, and you don't need a lot to make it happen.
And there are some that are part of a Miracle Grow like formula that's sold in like superstores for people to grow vegetables and stuff in. But he's totally like NASA will be very like cautious about introducing anything like that.
The internal makeup of what goes inside the root cylinder was actually determined by the life sciences group at NASA. So the makeup of the arsolite, the makeup of the capillary mat as well were determined by them, because I think they wanted to utilize what has already been flown just to make sure that there's no no component that'll cause an issue. But some of these things are all great ideas. If you can manage also the amount of available space so that you are giving the opportunity for the plant to take not only the liquid from the medium, but maybe even the oxygen that fills up in between them, it could be interesting. because the root cylinder was specifically designed to allow the roots as they enter or as they expand out of the root cylinder to utilize the balance of liquid as well as the oxygen too.
It would be fascinating to see some like X-ray CT models of the root system architecture after flight and of the terrestrial ground controls, like the version 4, round 4. Like so when they start talking to you about what to do next, you should say that X-ray CT. You can scan it in situ or go straight through, and you'd be able to see a three-dimensional model of it. You'd assume that the water would create a certain pattern based on the environmental constraints. So you can imagine having a ground control where you change the amount of oxygen or the amount of water or the amount of nutrients, and then like going through all the different possible combinations and then comparing those three-dimensional structures to those produced on the space station version. And then you'd be able to go, like the space station version, because of spaceflight and microgravity, it looks like it's nutrient deficient, or it looks like it's flooded, or
it would be interesting to install a couple sensors, the way that you've described, and I think obtain more data off of the experiments. I agree.
Mm-hmm.
I'm just such a nerd. I love it. The stuff you work on is so cool. It's like, it really is the cutting edge. It's the holy grail of astrobotany, really. The capacity to be able to have like a self-watering pot with minimal user interface, and that won't be knocked out by intergalactic cosmic ray going through its processor. It's a it's a difficult thing to crack, but it's going to open up a whole new era of space agriculture. It's very cool stuff.
Cool.
We love it when you are a nerd. Awesome.
I think that's 24/7.
That's probably everyone on this call.
Exactly. Other questions? I didn't have a chance. Let me check YouTube real quick here. Is that material sodium polyacrylate? Jim Cottrell asked that.
The material that Richard was talking about, you mean?
Yeah.
Yeah.
Oh, never mind. It says he's just answered himself back in chat.
Okay.
Jim probably should join us on the inside here. He's a teacher in Yucca Valley there in Southern California. He's looks like he's in YouTube instead of in here. I'll send him a link. Wonderful, David. Thank you so much for the background information and the research that's required. I guess hoping for that Goldilocks there. Maybe too many cooks in the kitchen, but maybe version 4 will get you what you need.
We'll see. It's been a year between iterations. Hopefully we can move faster than that.
Now, so one thing, clinostats or things like the vomit comet, these that you go into microgravity. Obviously, those are just a few moments in time, and the clinostat probably really doesn't give you an effective result. But are you doing other simulations in microgravity? Is it really got to be on station?
So PONS 1.0 was originally designated to fly in a parabolic flight. So on the vomit comet, but that contraption only has something like 20 seconds of weightlessness, right? And I think NASA had determined that it wasn't enough time for them to do all the observations that they wanted to do, so they decided, let's just send it to station. So we lucked out, actually, because we got put on a rocket earlier than expected.
And the fluid dynamics also, I'm curious about what kind of modeling is done when you look. I imagine you're using supercomputers and what have you. And this gentleman from Oregon, what kind of modeling tools they might use to think about what the adhesion and cohesional properties that are and how they're changing, how water changes in microgravity.
So he has actually designed a chamber within his university building that it's about as tall as he can go, but in comparison, there's not much room, for something to fall. But I think he was telling me that it gives about 2 seconds of weightlessness. You know, so for him, that was enough to give some pointers in terms of, you know, how much surface energy was needed, how much, you know, capillary mat in order to channel the water, you know, in the ways that we were looking for. So it's interesting. We actually had him visit us, and he used to be part of NASA before he took over that department at University. I think it's University of Oregon, but you know, he spent about 2 days with us at Tupperware, just specifically, you know, working both in terms of evaluating prototypes that we had and calculations in order to come up with, you know, what he thought was going to be the right makeup. Because
cool.
we played a lot with geometry as well.
Yeah, his coffee cup's pretty incredible.
His coffee cup is really incredible. So he showed us a lot of movies of astronauts using his coffee cup, sipping from it, and then tossing it to the next person. You know, and the thing would go and tumble through the space over there. The other person would catch it, and he wouldn't lose a drop. It was amazing.
Yeah, just understanding how fluids operate. There's been a couple questions about different vehicles or opportunities to test ponds. Laura, you had a question, and I think Liam, you also had a question. You go ahead and feel free to ask.
Sure. So I was just thinking if it wouldn't necessarily give enough weightlessness on the Vomit Comet, what about doing some testing, even with the ExoLab, on a high altitude balloon?
It's all interesting. I mean, I think that there's a lot of opportunity to test it. In the past, we just went with what NASA thought was necessary.
Oh, there you go.
The high altitude balloon, the best way to do it would be on the South Pole, because you couldn't get a weightlessness effect, even if you dropped it because of the wind pressure, you wouldn't have much of a fall time. But on the South Pole, where the magnetosphere comes down, there is an NSF balloon project that they put balloons up for about a week and they fly around in the high atmosphere, like exposed to elevated amounts of intergalactic cosmic rays. They often put like telescopes on there to look out at the stars because you get less resistance because of the lack of the magnetosphere. But it's a great opportunity to get an increased amount of intergalactic cosmic rays, which is one of like the showstoppers for the journey to Mars. And being able to understand how life responds to that exposure is really important. And so having plant growth equipment that is modulating like this, that will keep them and allow you to grow the plants fully moistured, fully watered for about 7 days, 8 days as it goes on this high altitude balloon flight, would provide us the opportunity to be able to then look at the plant's genetics and physiology and see how it interacted with those the high altitude balloon environment with those increased galactic rays. So I love the idea of the high altitude balloon experiment. I've been dreaming about it for a long time, but the hardware to grow the plant and keep it up there just doesn't exist at the moment, but the balloon goes up every year.
There you go. Well, I'm packing my bags to McMurdo Station. Let's do this.
You got a chance for one more question?
Absolutely.
Yeah. Sorry. So this is Liam. I'm here. Yeah, the question about using your experimentation on the Vomit Comet had me think about another microgravity environment that is not quite the same as in orbit, but it's suborbital. Have you considered utilizing what's available with the Blue Origin, New Shepard, suborbital flights where you can get about 4.5 minutes, maybe even a little bit more, and they have an experimental platform sort of way of having people engage with that.
So that's an interesting idea, Liam. I think that up to today, those flights have not really been available, and I think that maybe it's something to look at for the future. You know, the interest of NASA to send it straight to station, I think for us is good because then it actually gets a couple, you know, lengthy durations of testing. But I think in the event that they don't have any availability, you know, that could really be an opportunity.
Yeah. And Ted, do you know anything more about that? Because Erica, that there were various,
yeah, in fact, I want to
think,
get her
test examples.
I want to reach out
where-
to and get Erica in from Blue. That 4 minutes is not a lot of time, but I guess if we think about from a design practice, David, one thing that could happen if you've got those 12-month gaps, you can run a series of prototypes in parallel and look for results.
Actually, 4 minutes is quite a lot of time. If you are actually looking at how you are progressing or transmitting water, transporting water, actually, I think is the right word, and see how things actually wet out. 4 minutes can probably give you what you're looking for.
Well, there you go. We ran we were lucky to kind of be a stowaway on a NASA sounding rocket a number of months ago. We were stuffed into the nose cone. We weren't doing plants. We were running simulated satellites. But it's nice to get up to 400,000 feet and get 4 or 5 minutes of microgravity.
And by we, you're doing the royal we, right?
No, well, that was Magnitude.
That's right. Yeah, it was the Magnitude. That's right.
Yeah,
It was the equipment, not you and Ted.
Not me.
Not you and Tony.
Experiments was really fascinating. It was one of our original engineers, and he wanted to look at early planetary formation. So he had salt crystals in a chamber with cameras on it, and the idea is, would the electron, I guess, attraction of the salt crystals as they got into microgravity actually start glomming together, forming kind of a proto-planet, if you will. And so that was an interesting concept that he had a chance to explore with just a few minutes of microgravity. So that's kind of neat. Liam, thank you for the view of Earth there. Look at that. And that's a recorded one, I guess. That must be.
Oh yeah, it's a recorded one.
Yeah.
By the way, yeah, your experiment, the Ponds experiment, is going to be passing by all of us in Southern California in 23 minutes.
Oh,
so
cool.
We'll have to check in on that.
Yeah. The video I was just showing there was from my friend Sean Doran, who creates real-time renderings from astronaut to time-lapse photography.
Really cool.
So, anyway, just to explain
Very,
what was
very
going on there.
Awesome. So I was looking, is Beth on from Leguminaut? I don't think I see her on, and maybe I got something closed here. Our next section has been planned for another culinary expedition from an Orlando vegan restaurant called Leguminaut. We thought it was appropriate, considering this is the Leguminaut Challenge. And last week they had a nice little recipe for us, and I thought they were going to show up this week again. Maybe I've got my hours here wrong, but if they do pop in, we'll jump over to that. But David, thank you so much for sharing the research that you're doing and the deep engagement with NASA, and probably more importantly, to reacquaint us to what the research is happening with Tupperware for sustainability and for food preservation, which I think is super critical, especially considering not every home has refrigeration around the world, and how we can optimize food, especially, well, geez, 40% of our food.
We have to work on both sides, right? On the side that provides food, but on the other side, which requires, I think, a behavioral change in terms of what we do with food.
Right. And well, my goodness, with the pandemic really affecting all of us around the world and all the different countries that are joining us today, supply chain, right? Toilet paper was the first thing that appeared, which was kind of weird, but the food, and now we're talking about our meat products. And so as this gets disrupted for various reasons, not enough truck drivers or not enough people, in the field harvesting the crops, what happens to our supply chain, and how can we preserve the food from farm to kitchen, right? So that's a really intriguing question. And of course, if you're on your way to Mars, you better make sure you got your vegetables nice and crispy, and preferably you're growing your own, right? But very, very interesting. So I think I guess Beth isn't on. So what we're going to do is just maybe do more of a general conversation before we get into our next segment and talk about, is Michael Wilkinson here with us, or is he in class?
Oh, Beth is here.
Oh, is she? Yay, let's bring her on. Pop back in. Where are you? So welcome.
So Ted and Tony, I'm going to jump off, okay?
David, thank you so much. I'll send you a note out.
Thanks, everyone.
Thank you, Dave.
Okay.
Have a great weekend.
Superb work. Thank you.
Take care. Bye.
So where's our friends with Leguminaut?
Yes. Hi. Good to see everyone.
Awesome.
I've
Great to
got,
see you.
I've got another legume for you guys, and one of my favorite recipes. I recorded a video of me cooking it so that you can see the whole cooking process. But
Yes.
we're gonna do lentils this week.
Okay.
And because I don't have enough weeks to do give every single legume a week, this recipe you can also use black-eyed peas for this recipe.
Oh.
I'm gonna be using lentils, but you could substitute black-eyed peas in the recipe.
There you go.
So
Showing some love to our cowpeas out there. Good deal.
yeah, I think you guys are really gonna like this.
All right. Now, our local resident lover here, Steve Jones, is not with us today. I think he's doing some distance graduation celebration with his high school senior. But go right, it's all yours.
Alrighty, I'm gonna go ahead and get my video up for you guys.
Cool.
All right.
Now, David is with Tupperware, and they're just down the road from you there. I think they're outside of Orlando.
Oh, wow.
Yeah, so he's with Tupperware, and I don't know if you caught most of his presentation, but we'll have to find a way to get you guys together.
Absolutely. Alrighty, so let me get this video going for you guys. All right, is that playing for you guys?
Yep, and you just paused. Yep.
Hey everybody, it's Beth here from Leguminati. I am here to show you another one of my favorite legume recipes. This one can use lentils or black-eyed peas. And it's a lentil sloppy joe recipe. So it's really quick and easy. Most of the ingredients, almost all the ingredients are really common to have in your pantry, and you can substitute if you don't have them. I'm going to show you how to make it really quickly. So the first thing that you're going to need for this recipe are lentils. And you can either get canned, like already cooked lentils, or dried lentils. If you get the dried ones, get green lentils. The red ones are gonna be too soft for this recipe. So I love red lentils, but for this recipe, use green. You could probably use black, like beluga lentils as well. And again, really, it's up to you what legume you want to use, because you can also use black-eyed peas if you want. So if you were using dried lentils, cook them according to the instructions. I think it's the ratio is 2 cups of water to every 1 cup of lentils. So, and I will make sure that I put that in the recipe to be 100% sure. But I think it's 2 to 1 ratio for lentils. And you're gonna boil your water, add your dried lentils. Green lentils usually take about 25 minutes until they're soft, 20 to 25 minutes. So not super long. And once you have your lentils cooked, the whole rest of the recipe comes together really easily. And if you're using canned lentils, they're already cooked. So it's really, really fast. So don't want to burn my onions. So the first thing that you're gonna do for this recipe is chop up an onion. I used like a sweet Vidalia onion, and I just used half of it. But you could use a white onion or a yellow onion, or even a red onion, whatever you have in your pantry. If you don't have an onion, you could use then garlic. You just want some of that like flavor going on. You could add garlic to this recipe as well. I just happen to be really low on garlic and I'm saving it for another recipe. And I don't want to go to the grocery store because pandemic. So otherwise I would totally add garlic to this. It's one of those recipes where you can be super flexible about what you're adding. So you're gonna take your onion and add a little olive oil, 1 tablespoon of olive oil to your pan on the stove, medium heat. And you're gonna cook your onion in your pan for 8 to 10 minutes until it gets, it's got a little bit darker than I normally do, but I kind of like that like toasty brown onion. So you're gonna saute it until it's nice and golden brown. If you want to go a little bit darker with your onion for that like toasty flavor, especially if in this recipe, because it's a sloppy joe, like the smoky flavor is really good. So you can kind of do that with the onion by toasting it to not quite burning it. You don't want to burn the onion, but you want to get it like right before it burns. And so pay attention to it. Don't film a cooking show for a Zoom meeting while you're sauteing your onion, because you might get distracted and it might get a little too burned. It's fine though. So anyway, we have our onion and then we are gonna add our lentils. I'm using canned lentils just because I have these sitting around, and this is about 1 1/3 cups of lentils. You could use up to 2 cups of lentils if you're making them dry. And it's just gonna, you're just gonna have a bigger batch. So I'm gonna, my lentils are leaking a little bit. So sometimes I add up to 2 cups of lentils to this recipe, and it just makes a bigger batch. So I'm gonna go ahead and add my to the pan and stir them all around. And then I'm also adding a can of tomato paste, something we all usually have in our pantry. If you don't have tomato paste, can of tomato sauce, even ketchup will work. And I know you say, oh, ketchup, that's weird, but ketchup will actually work really well in a sloppy joe recipe. So 6 ounces of tomato paste, like 1 can, or half a cup of tomato sauce or ketchup, if that's what you're working with in your kitchen. I like the flavor and texture of tomato paste, but you can totally substitute it if that's what you're working with. So then you're gonna wanna take this and like really stir. I'm using a silicone spatula, but you could actually probably use a fork to really get your tomato, if you're working with tomato paste, it's kind of thick. And so either like a whisk or a fork to kind of like get the lentils incorporated into the tomato paste. So stirring that up, it smells really good. And then I'm gonna add my flavor components to this. So when we're thinking about black eyed peas or a sloppy joe, like a lot of people like to add bacon. And I remember someone mentioning that last week. Ooh, I love bacon with my black eyed peas. But if we are growing our food and sending it into space, then animal products are going to be really hard to do that. So we are going to want to think of a plant-based option that can replicate the flavor of bacon. So bacon is salty, smoky, and fatty. So I like to add soy sauce because it has a salty flavor and also something called umami, which is a Japanese word for, it means savory. And so soy sauce has that really strong savory flavor. And so do a lot of animal products, a lot of meats. So when I am making something that's meatless, I really like to add soy sauce to give it a little of that meaty flavor. Another ingredient that's has a lot of umami flavor is mushrooms. So maybe some sauteed mushrooms would be really good in this too if you had some. That would be another thing you could add. Okay, so I got my soy sauce stirred in, got my tomato paste. So now I want to add something smoky. So my favorite smoky ingredient is smoked paprika. I'm gonna add a teaspoon of smoked paprika. It smells really, really good. I love adding smoked paprika to things that have tomato sauce in them. I think it's a really good combination. This smells so good. So now we're stirring in our smoked paprika. Now we're almost all done, but sloppy joes, the flavor of them typically has a sweet component as well. We have our salty from the soy sauce, our umami from the soy sauce. We have our smoky smoked paprika. We want to add some sweet, and you can really add any, there's a lot of different ways you can sweeten this. You can add brown sugar. That's a really common one. So if you have brown sugar, if you don't have brown sugar and all you have is white sugar, you can add some of that to it as well. If you added ketchup instead of tomato paste to your recipe, ketchup already has sugar in it. So you don't have to add any sweetener if you're doing the ketchup version of this because it's already really sweet. So my favorite thing to add is maple syrup. Not everybody has maple syrup. It's also expensive, kind of like a luxury ingredient, but a little bit goes a long way. I'm only adding 1 teaspoon of maple syrup. If you don't have maple syrup, brown sugar, or just regular old white sugar works. Molasses is really good too. If you have molasses, you can add that. So now we have our sloppy joe mixture. It's looking really good. I'm gonna go ahead and put this up.
You need smell-o-vision.
On medium heat until it just starts to bubble, and then turn it down. If it gets too thick and pasty because you're heating it up, so you're evaporating the moisture out of it, then you can add a little bit of water back to it. So I would just add water 1 tablespoon at a time because you don't want to make soup or making a sloppy joe. And I like to err on the side of making it a little bit thicker in texture than too thin. So while you are waiting for your sloppy joe lentil mixture to come to a bubbling simmer, you can cut your buns or rolls if they need to be cut in half to put your sloppy joe mixture on. You might have bought hamburger buns that are already cut in half. If you don't have buns, you can use bread. I like to toast it before because it makes it a little sturdier to put the sloppy joe mixture on. If you don't have bread, you could make rice and put your sloppy joe mixture over it, or It's really good on top of mac and cheese, believe it or not. So if you have some leftover mac and cheese, put your sloppy Joe mixture on top of it. It's really good. So I'm gonna go ahead and cut my roll. So, and then if you really want to make it really yummy, you can warm up your bread. Some people like to put, you know, like fixings, like slices of onion, or I don't think I don't really like to put a anything other than my sloppy Joe mixture on this. But if you do, if you have a preference, go for it. Jalapeno slices, if you want it spicy, get creative. You could put cheese, if you have vegan cheese on top, that would be good too. So my mixture of the lentils is already starting to bubble, and that was just a few minutes. So of course, the longer you let it sit and simmer, the more your flavors are gonna incorporate. So I would give it at least a good 10 minutes before you put it on your, before you serve it on your rolls. So this is, at this point, I would turn down the heat to low, and I will come back in 10 minutes and we will enjoy our sloppy Joe.
All right, so that was the cooking component. I'm going to share the taste test with you guys next. All right.
That's not fair.
I know, it's just really
Next week,
not
they send
fair.
us all via overnight delivery. The rest.
Exactly, it's just cruel.
Now you guys are based in Orlando, right?
We are
And
based.
you got pickup now, so you are kind of open.
We are doing curbside pickup, and then we're doing Uber Eats as well. And then over the next couple weeks, Florida is going to start opening up, and we'll be able to have outdoor seating. We are not having, some restaurants are already going in Florida are already going to start to have indoor seating. We will not be doing that. So we're highly encouraging everyone to do curbside. That's the least amount of contacts, or Uber Eats. So we're hoping
all our friends there visiting Kennedy or coming through Orlando, drop by.
Absolutely. All right.
I volunteer to come and do a taste test on your patio seating.
Lori, that'd be great. Yeah. I'll be there tomorrow. I'm there every Saturday. So if you come on a Saturday, make sure you say hi to me.
I'm just wondering what these sloppy joes would do in microgravity. Might be a little messy.
I know. Well, so we do have our sloppy Joe at the restaurant, and we put it inside a crunch wrap. So it's wrapped up in a tortilla, and it's like encased.
Oh.
So that will actually make it really like more contained.
Okay.
Because a sloppy Joe in space just sounds like a nightmare.
Very, very sloppy.
It does. All right, so let's go ahead and I'm gonna get my taste test going for you guys.
Good deal.
Alrighty. Okay, let's see. Making sure this is not the cooking version. Nope, that's still the cooking. Don't want to give you guys the cooking demo again.
That's okay.
They look very similar, so I'm just trying.
And also, we're getting your recipe from last week as well as
absolutely. This week. Yeah, I'm gonna post and make sure that you guys get every recipe that I share, and along with, since I'm doing substitutions, the recipes will have ways to make it, if you don't have, especially right now, since ingredients can be scarce for some people, like I was talking with the ketchup, that way it'll be friendly for everyone to be able to do it. I don't want to have everyone going out and getting strange ingredients. All right, so
that
let me.
smoked paprika is not strange.
Right, that one.
That's an essential, right?
Yeah, that one I find like everyone should have. That one's a really important one. All right, so.
Now let's make this a global challenge. Also, you've got a favorite legume recipe. We want to hear about it.
Absolutely.
You know, as we prepare the right legume to send to space, we want to know what tasty things you can make with your legume. So we've got our lead here with Leguminaut, specialists in legumes. But you got a favorite family recipe or one you really love making, or you heard about, you want to try? Share it with us. Let us know.
All right, I'm gonna go ahead and play this for you guys.
Okay, so I simmered my lentil sloppy joe mixture on the stove for
For some reason, we're not seeing it.
medium heat, and everything's all nice and incorporated.
Beth, it looks like we're looking at your downloads folder, but not the video.
Oh, okay, hold on. I am new to sharing files on Zoom. So you guys are my first Zoom guinea pigs with sharing files.
There you go.
It is.
I think it's just the screen you choose to share, I think is what you're.
Oh, okay. Yep, there we go. Is it coming up now?
No, so maybe stop sharing and then reshare.
That's what I'll
Make
do.
sure you're selecting the video. Yeah, you got it.
Thank you guys for your patience with this.
Oh, totally cool.
And while we're waiting for that, the view that I'm just showing you as the station's going over Ted, Tony, and myself. Anyone else on the West Coast?
Fantastic.
Anyhow, it's going right down from north to south. So
wonderful.
Yeah, and Javier, that website which is behind you, which is really awesome, is not currently on the live view.
It's on YouTube.
Oh,
Oh, there you go. They've just switched it.
There you go.
Awesome.
All right, back to the regular program.
A little sweet. That just smells really good. So we're gonna go ahead and take our sloppy joe mixture and put it on our roll. And I'm gonna go ahead and give it a taste test for you guys and tell you how it is.
This is very cruel, Beth.
Yeah, it is.
It's really good. Really, really good. Really flavorful. I feel like this would be a really family-friendly recipe. So I would love for you guys to give this a try. I would love even more for someone to try this recipe with the black-eyed peas instead of the lentils. It's gonna have a little bit of a different texture. The black-eyed peas are bigger. So it's gonna be a little bit different, but the flavors of the smoky and the umami are gonna be really good with black-eyed peas too. So if anyone wants to give this recipe a try, I will make sure that I write this one down as well, and give it to everyone. So now we're gonna go live for some questions and some more information about lentils and black-eyed peas.
Awesome. Thank you, Beth.
All right. So that was a really fun one for me to make, and it's very, very easy. And like I said, it's just such a quick and easy thing to do with lentils. And most of those ingredients are really common. So I loved, it was inspired by the lentil burger recipe that we talked about last week. I was like, I love that, but making burgers is a lot of, it's a lot of steps. And this would be a fast one, especially for like a beginner cooker or someone who's never made a vegan recipe before. Yeah, this is, if you've never made lentils before, this is a really good starting place. So, and like I said, maybe you don't have tomato paste because you don't make tomato sauce from scratch, but you have ketchup. You can totally substitute. And that's why I love this recipe, because it's such an easy one to do. Another thing I really like to do with lentils that is, again, not so space friendly because it's spaghetti sauce, but I like to just have either spaghetti marinara sauce that I made, or you can buy store-bought, and I put lentils in my marinara sauce, and then serve it with pasta. And it's so easy because it's not even a recipe. You're just adding lentils and you're adding that nutrition, because lentils are very, very high in protein and iron. So they're a really good one when you're trying to replace the meat. Adding lentils is a really good one to sub. And I find them very filling. And my 5-year-old loves lentils. So they're actually pretty kid friendly and good for picky eaters, because they kind of cook down and almost get like, they get into the sauce, and they're not so they're not so scary for someone who is new to eating legumes. So.
Good deal. All right, Javier, you have a question?
Well, not a question, but a comment. This is good because, in my country, in Mexico, we just have one way. I don't know, I lived for many years there, but we just have one way to eat lentils, which is like the soup. I don't know if you ever tried. It's like a soup with onions and tomatoes, and that's it. No more. So this is a different way. This is cool because you're learning new stuff here.
Awesome. Yeah, I love lentil soup. And then in India, red lentils are really common. And lentil flour, which again, if you're thinking about bringing lentils into space, whatever legume you're going to be using, you're going to want to turn it into multiple things. So you're going to want like the whole legume, but then also making flour out of it. So lentil flour can be used to make this really, really delicious flatbread that you can get at Indian restaurants, and it's really, really good. There's also on the market, like at the grocery store, there's like lentil tortilla chips and things where they're adding lentils to it. And so adding a nutrition component to something, usually tortilla chips just have corn or wheat flour in them. So if you're adding lentil flour, you're making something more nutritious that is a food that is commonly eaten. So I really like the idea of incorporating legumes into things that they might not necessarily normally be in, to make that more nutritious. So if you're eating tortilla chips and they have legumes, then they're going to have more protein, they're going to be more filling, they're going to have more micronutrients. So that's definitely some forward thinking ways to incorporate things if we're going to bring them into space. So making sure that we're getting all those macronutrients and everything, but not necessarily eating it as a sloppy Joe. Well, it's really fun. It might be easier to eat it in a different way. So.
Well, I like that wrap method. That sounds pretty good.
The wrap is, so we, right now at Leguminati, our whole menu are these crunch wraps. I showed a picture of one of them last week with mac and cheese inside of it. And so we just take a large flour tortilla, and I'll have to show you guys how to make them because they're really fun. So I'll do a demo on that, because you wrap it up like a present. And so you put all your ingredients inside of it, and then you wrap the tortilla and press it like if you were making like a grilled cheese or a panini or something. And so everything is all contained inside that crunch wrap. We cut it in half so that you get 2 semicircles and you can eat it. And it's really, I can eat mine while taking care of my 5-year-olds and like running around. It's nice to have a food that you can like hold in one hand and like run around and like, so I'm like, okay, I'm not in space, but I'm chasing my kid. So it's like, it's a really portable food. And you can put so many things in it. So that makes it really fun.
Awesome.
Yeah.
Anyone have some thoughts around that? Maybe a challenge? You have a legume that you'd like to see a special recipe made for?
I'm just super hungry now.
I know, right? I can actually kind of smell the the sloppy joes. I'm ready to eat. I haven't had breakfast yet. Just like 3 cups of coffee. So,
I made the sloppy joes on Wednesday night, and we had leftovers for not very long. I had made a big batch, and it was gone very quickly.
Well,
So yeah.
And so this is super fun and important. When we think about doing experiments in space, sometimes it gets very heady. We're thinking about the protein expressions and what the roots are doing. But at the end of the day, we're talking about food. And probably one of the most special places on the International Space Station is the galley, where they're working, and they've got more than a dozen sunrise and sunsets every day, I think 15 or so. And so it's really weird to your sleep cycle. And you're in these artificial lights. You might have a chance to look at the Earth out through the cupola. But where does life kind of get normal? It's in the kitchen, where you've got your crewmates there. And it's really kind of fun to watch how they prepare their meals. There's some interesting videos we should probably show maybe next week as a prelude to what you're cooking, and just to think about what an experience might be in a galley or in a kitchen where you're just kind of floating around. Right? It's like a little different experience, and you can imagine what the heat's going to act differently. So you can't really pan fry anything or things like that. It's like, wow. But we're here on Earth, and we're thinking about food and food sustenance. So definitely, all these amazing, cool recipes that we can make with our legumes is really fun. So thank you so much for sharing that with us, Beth. And what we want to be able to do is, even your little video that you shared, we didn't do a cooking demonstration for last week, but maybe we can take that, do a little editing, and get that in as part of our media, and make sure people can see that as a little breakout session. Would be kind of fun. So very cool. Thank you for joining us again this week. And we're looking forward to, do you have an idea? Can you give a little hint what's happening next week as far as recipes?
So I really want to get something sweet for you guys. And my favorite legume to do for desserts is chickpeas. Everyone finds it really unexpected, but I'm going to show you some cool stuff that you can do with chickpeas. And then we're going to talk about some of the things I do with chickpeas you couldn't do in space. And so we're going to talk about like what would be possible and what wouldn't. So that'll be really fun. I can't, like, now that's kind of where I'm at. Like when I'm cooking, I'm like, okay, like this wouldn't work in space, but the flavors can. And that's what's cool, is like, you couldn't make the sloppy joe the same way, but you can have that umami, that salt and that smokiness, and combine them together with a different cooking style to get the flavor of the sloppy joe. And so I think that is a really fascinating thing that could be brainstormed, is like, how do we get these flavors so that we have a variety of tastes to eat? Because it really does help morale to have different flavors and not be eating the same exact thing every day. So
exactly.
Well, and Beth, keep in mind that ultimately going to Mars and setting up those first explorers and a colony, that they are looking for the range of things that they can do with plants. So while it may not be possible to cook some of these things on the International Space Station, on Mars, you do have some gravity. And so you could do some cooking, and chickpea cookies would sound mighty tasty as a good alternative to eating prepackaged food that's been, in these plastic packets for 5 years. Right. So I think that even though it may not be able to happen while you're in transit, once you get there, you'll have more cooking options on when you're on the surface.
That's a positive attitude. That's the way to look at it. Can't wait to get to Mars. I got chickpea cookies coming.
Exactly. It's something to look forward to. It'll make you happy. Go cookies.
Someone hit the gas, right? We need to get there a little faster. Are we there yet? Wonderful. Thank you so much, Beth.
You can start growing your chickpeas while you're in transit. Then by the time you get there, you can harvest them. And
there you go. Perfect. So that's what we have to do. We have to find the right legume, though, and I'm going to segue to that. And just as a note, at the top of the hour, in about 15 minutes, we're going to segue away from talking about space and legumes, because we have another balloon launch, this time coming out of Southern California with YWCA. Some young gals might be joining us already, popping in, and Liam is going to be kind of commanding that adventure. So that's coming up in a few minutes. But why are we here? What are we doing? The Leguminaut Challenge is a chance for us to get together at a planetary level to think about food and zero hunger. A UN Sustainable Development Goal. We heard from David Kasuma a little bit earlier, the World Design Organization. He kind of laid out those 17 initiatives that the UN has. And there are people on the planet that aren't getting a meal. 1 in 5, apparently, according to the UN, are undernourished, and that's a significant amount of people. So, can legumes pave the way for sustainable living for all our folks? A big challenge with modern agriculture also is the way we're depleting our topsoil. According to the UN, we have about 30 years to 60 years left of topsoil. We're depleting it very rapidly because we're pushing it as hard as we can with pumping it with artificial fertilizers, and really getting the most production that we can because we have so many people we need to feed. But our systems are inefficient. As David shared with us earlier, 40% of our food here in the United States is getting tossed. And so those are pretty massive compost piles, but wouldn't it be nice to get in the bellies of some people that need it? So legumes are a great storage of energy medium. If you think about it, dried beans or what have you can really get in a bag or a sack and get sent somewhere else. But what's also nice about the legumes is when they work symbiotically with a bacteria called rhizobia, they help put nutrients back in the soil, nitrogen specifically. A nitrogen-rich soil really helps replenish your topsoil and helps us have longevity. And so these are some things we're exploring. What is the right legume that we can send to space for maybe a garden on Mars, but also as we're taxing these little plants, putting them under stresses, how might they change? What kind of expression is going to happen? And then I think if Dr. Barker's still on, he can dive in much deeper than I can. I think he's jumped off already. But if anyone else wants to jump in and what they understand there, I think this is something that's very interesting to us as we look for perhaps some species or types of plants that could be able to withstand drought, so they're drought tolerant. That maybe they might produce a higher yield for the amount of nutrients that are required for them or amount of water that's required. Maybe they can grow in a brackish water in a kind of a delta where you have salt water mixing with your fresh water. And so all these things can help us determine not just this really kind of cool adventure of getting these settlements on other worlds on a surface of our moon, but also providing a nutrient food source for just the inhabitants of our lovely planet. So this is really exciting to us. And the Leguminaut Challenge is open to anyone. There is no requirement to purchase anything at all. We just need your feedback. We've got this phase 1 is ground trials in which we want you to grow a legume. We have 9 that we've pre-selected, but maybe you've got a favorite one you want to try. We want to hear about that. You can go to our website, Magnitude.io, select the Leguminaut Challenge, and go ahead and enter in, log in. There's a really easy way if you're a parent and get your children involved, you can do that. It's really designed for teachers based on our educational system. This experiential learning, project-based experiences. Let's start with something super simple. Let's look at the origins of life. Let's watch a plant grow. Gil from Japan kicked off our session today in which we were actually looking at the effects of gravity. And so you can kind of do some interesting science experiments with just a simple seed. But whether you're living in an apartment, you've got a little windowsill in your kitchen, you can grow some legumes in, or maybe you've got a nice big garden in your backyard or side yard or your front porch. Let's find a place where you get some nice light and grow something alongside with us as we explore what might be possible. And we're running this now through, I keep on getting my dates mixed up, of course, sliding them back and forth, but I believe by the end of May, we're going to need to select, and it might be extending now. And a lot of it is we have to connect with our flight criteria. Just like we're concerned about a sloppy Joe in microgravity, and we're going to have to get a plant that grows inside a certain volume. In this case, we've got about 20 centimeters by 10 centimeters by 10. Essentially, 2 of these is what we're going to be growing our laboratory experiment in space in February. And so help us determine what we want to put in our lab. Here on Earth, if you get that far with us and you want to do something in space alongside our experiment, Exolab is what we use here on Earth. Hundreds of schools around the world, whether we're in South Africa or Germany or Japan, Canada, Mexico, here in the United States. We've got other countries joining us as well. Kenya is not here today, but they're involved. They can run a laboratory experiment here on Earth while it's connected to a sister experiment in orbit. And that's February, so you've got some time, got about 6 months to help us determine what plant we want to put in here and grow here on Earth as well as in orbit. And there's some other experiments we're going to be doing as well, like what is the right material. David Kasuma from Tupperware, there with the pond system on the veggie, is actually working on wicking systems to really provide the right level of water and nutrients to our plant. Can't overwater it, can't underwater it. And water behaves completely differently when you don't have much gravity. Here on Earth, real quickly, is the ExoLab. It does things for us so we don't have to worry about it. If you're growing your own plant on your windowsill and you're part of this experiment, we want you to observe it every day. How many leaves does it have? What color is it? If you're growing it in some soil, is it dry or damp? Just kind of measuring the height and what have you. All these observations are good. ExoLab does a lot of that for us and adds some other measurements as well. So it measures carbon dioxide. And now in our, like, I'm here in my garage, thanks to the pandemic, but here in my garage is probably like 1,000 ppm because I'm breathing in here and I don't have many plants. I've got a couple plants growing, but not as many enough to kind of absorb that CO2. Outdoors, it's around 450 or so. It's getting kind of high out there. We worry about global warming, but it measures ambient conditions. Also temperature and relative humidity. So we get a chance to see those diurnal cycles as it warms up and cools down. It even measures the light levels. This device lights up and you can change the frequency or wavelength of light. Plants behave differently with different wavelengths. You can see Michael there is logged on. He's all lit up in a magenta light. Early growth for plants, they love that magenta. And I think Michael's growing right now, come to speak of it. Or you could have like a white light or a red light. How might that affect your plant? So you can adjust the lighting wavelength, its intensity, how many micromoles per square meter is light hitting your plant, and also the photo period, how much light over what period of time. So you might have on the space station, we run a 16-hour photo period, 16 hours of light, 8 hours of darkness. You might see what it looks like with just like 4 hours of light or no light at all. So the nice thing about the ExoLab, it's running all those measurements for you, even if you're not there. So we have some classrooms that are still running their ExoLabs, even though no one's there. And we can log in and we can see because it also captures a picture once an hour. And I think especially for our younger students, watching a plant grow, it's kind of like watching paint dry. I think they love it when they can see a time series when it's in our libraries or museums. They usually pop in once a week when we do have school, and they love to see the changes if they can remember. But with something that's capturing all the information, it's much easier. And from a research perspective, just to cap it off, we have the ability to look at all of these variations, different temperature, different humidity, different light levels. And by the time we get ready to fly the same plant, and we now have a crowdsourcing platform in which we've got all the imaging, other information. We hopefully have some observational data by parents or students or even citizen scientists. If you're excited about ExoLab, we haven't really worked with too many DIY kind of citizen scientists, but you can join us. Come aboard on ExoLab number 8. It's our 8th mission to the space station. So we're really excited about the work we're able to do up there. And one thing I haven't shouted out is without the National Laboratory, I'm going to turn this over to you, Michael, since you popped up a screen here. But without the U.S. National Laboratory on the space station, we could not do these experiments. Funded by NASA through the support of Congress, we're able to do these experiments for students around the world. Our focus is domestic, that's the U.S. National Laboratory, but Magnitude enables us to, we can then reach out to all these other countries around the world, and we welcome you. If you're seeing us for the first time, reach out, hello@Magnitude.io, or just go to Magnitude.io and join the Leguminaut Challenge, because we really want to know what you think we should grow in space. And when we do grow that, working with folks like Beth and other creative culinary experts, what can we make? What kind of food can we eat? So Michael, why don't you walk through a little bit what you're showing us right now?
Sure. So this is the student portal, and it gives you a view of what you might see if you were running an ExoLab experiment. The frame on the left actually shows my ExoLab currently. We're just running a prototype now, so the experiments don't match. But I'm looking at clover, And those 4 tubes have actually been inoculated in 4 different ways. One of them is a control. Another is using a powdered inoculant that you can purchase commercially. Another is inoculated with an actual nodule that was produced by the clover plants. And the final one was inoculated with some tea made from the soil that those clovers had nodulated in. So I'm looking to see what the best in terms of the next round of nodulation is, because that's really our goal in this experiment in February. Is we want to see, can we get these legumes to nodulate in microgravity? That's the key to the whole renewal of the soil, the nitrogen fixation, and the real benefit of these plants beyond just a source of food.
Can you actually walk through the mechanics of that a little bit? Is it like a refresher? We cover pretty much every episode, but I think it's good to make sure for our first time viewers, what is this nodulation? What's really happening with the bacteria and with a plant like a legume?
Sure. So I'll stop sharing this. I think somewhere I have a diagram, but I'll talk you through it. So we're swimming in an ocean of nitrogen. It's the largest single constituent gas in our atmosphere, and it's inert. Nitrogen, the chemistry of it bonds 2 nitrogen atoms together to form an N2 molecule, and the bond between those 2 nitrogens is incredibly strong. And it takes a tremendous amount of energy to break that bond and release the energy that's contained within it. And that's what our bodies do at the cellular level, which is pretty incredible. So you're getting these little micro explosions inside the cells of your body in the mitochondria. So you're actually running on nuclear fission at the cellular level.
Fusion, not too good.
Yeah, fusion, not so good. That could be an interesting chimera. So that nitrogen, it's inert. It's non-bioreactive. It takes the amount of energy of a lightning strike to actually break those bonds apart, and that can happen locally in the soil. But if you think about, unless you're in Florida, we don't get nearly enough lightning strikes to sustain the nitrogen levels needed in the soil. So there's got to be another mechanism. And what has evolved are these bacteria that live in the soil, and they have the ability to actually take that nitrogen and use it to produce ammonia, which then can be converted into nitrates and nitrites. And that's the food that plants get. In your NPK, your osmocote, the N is the nitrogen, P is potassium, P is phosphorus, K is potassium. And why is it K? Because in Greek it's kalemia. So that's just answering my 4th grade question every time I say that. So the N is the nitrates and nitrites. That's the form that it's in. It's not a free nitrogen. And so to produce that, it takes a tremendous amount of energy. It takes a lot of fossil fuels. You have explosive byproducts. Really not a good day in a space habitat. Free ammonia in a space habitat, a droplet of ammonia that you inhale is deadly. So you can't be using ammonia and hoping that it'll convert naturally. Also, you're going to need some bacteria to do that. But there's this incredible relationship between legumes and a whole collection of soil bacteria, rhizobium, that do this as part of their life cycle. And it's a symbiosis. Both in a symbiotic relationship, both organisms benefit from that relationship. In this case, the rhizobium, the bacteria, are producing the nitrogen compounds that the plants need that then get passed through the entire food chain and allow us to have DNA and be a living organism, unlike those viruses, because they have to invade us to obtain that to replicate. So life couldn't exist without these bacteria. The legumes, in exchange, give the bacteria the carbon compounds, sugars, because plants are really good at producing glucose. That's what they do. That's their job. And what happens is actually the plants send out a signal into the soil. The rhizobium are attracted by that signal. They infect the roots of the plants. And in the proper conditions, that pathogen becomes a symbiont. And they infect the root tissue. It forms a nodule, which is like a tumor. And inside that, the rhizobium are doing their nitrogen fixation. So it's an incredible biofactory if you think about it at that level. It's fascinating to me, just the science of it, the biochemistry of it, just everything, all the evolution. And humans have been, we've been carrying around legumes and eating them and using them in all kinds of different ways, feeding our livestock for literally millennia. The ethnobotany, we go back and look at 10,000 years ago, we find evidence of lentils and chickpeas and mung beans in the carbonized in the soils that archaeologists have uncovered. So
yes, but were they making sloppy joes?
Probably not. At least not with beans. If they were making sloppy joes, they were throwing that critter on the fire.
Oh my goodness. Hey, we're at the top of the hour, but this is a great kind of overview of this nodulation that we're really looking to achieve. It's never been done in space before.
Right. Isn't that amazing? This
right?
critical piece of life. We've never been able to do it on orbit. So that's the exciting thing about this experiment. Teachers and students could literally be the first people to figure out how to do this and how to make cropping in space sustainable and how to make cropping on this planet sustainable. And if that doesn't excite you, I don't know what will.
Mm-hmm. Absolutely. So thank you, Michael.
You bet.
I think we've got 4 or 5 more sessions remaining for this season. This is all kind of a new invention that we put together at Magnitude based on this pandemic. We're all kind of stuck at home if we want to think about being stuck. But I think what's happened is it's created some innovation and creativity. I'm seeing in all the people we've been in contact with looking for something fun to do. It's pretty big project thinking about trying to feed a planet and doing some research on the space station. But you can do it and join us if you haven't already. Just go to Magnitude.io, sign up for the Leguminaut Challenge. But now for something really super kind of interesting, I'm going to bring it a little more terrestrial, but we're still going to get in the atmosphere here. It's just not going to be in orbit like the space stations, 250 miles above the planet, traveling at some ridiculous speed of 17,500 miles an hour.
It's 5 miles a second, 25 times the speed of sound.
And I've got a view of Earth behind me right now, thanks to ISS above. Liam.
If you look at Liam's screen, he's got it also. Look at the limb of the Earth. There, that thin blue piece. That's what Janelle's going to take us on a trip into.
There you go. And so I'm going to actually turn this over to our guest today at the top of the hour is the YWCA in Southern California. And, like all of us, what happens when schools get shut down? What happens to summer programs? What happens when we normally were able to sit around a table and do a project together? Well, it becomes a little bit more of a challenge. So we're putting our heads together. As you know, we're now launching a balloon every week. We've taken a little break from launching them up here in Northern California. And we brought a ham radio expert, Liam Kennedy, down in Monrovia, who's going to help some young women here. And they're going to share their knowledge. I think they're pretty fast learners. We just introduced this to them just in the middle of the week here. So with that, I'm going to turn it over to Janelle. Since you're joining us, Janelle joined us a couple episodes ago. JPL engineer doing some really cool stuff, but she's also board member of YWCA. So Janelle, I want to turn it over to you and maybe give us an intro. I see Jomi's on as well, and it looks like some of our students are here in addition to that. So I'm going to turn it over to you for a basic intro. We'll kind of make it up as we go along, but at a certain point, maybe half past the hour or 20 till, Liam, I know, is going to get in a car and drive off to actually get this thing underway. So the floor is yours, Janelle. Hello and good to see you.
Sorry about that. Always putting out fires.
Uh-oh.
Hi, everybody.
Fires at JPL.
Janelle,
hopefully
where are
not.
the outstanding pink
Yeah.
headphones?
What was that?
Where are those outstanding pink headphones?
Oh, see, I should have put them on. I have them. I could change over later on. So good morning, everyone. Thank you so much for allowing us to be part of your Friday meeting Zoom call. Ted, Tony, Liam, so we're so thankful for this collaboration that has been born out of this pandemic, because at the YWCA, our main service is to providing girls in our community with the inspiration and tools they need to feel empowered to have a STEM career in their future. And so when we switched over to this pandemic mode and we couldn't meet with our girls in person after school, the question became, how can we continue our programming and still make a meaningful impact and deliver our mission? And that's when I got talking with you guys. And this great idea was born of having a virtual balloon launch that symbolically could bring us all together during these hard times. And so with that in mind, we decided to have the girls kind of run the show here, because they're the ones who are learning and growing from this experience. And so I'm not going to be giving anyone a presentation today. The girls are going to be taking over and showing you what the plan is, what they've been working on, what they learned, and also letting you all, the audience, ask them any questions about this experience for them and give them a chance to talk to you about that. And they're also going to be calling the shots on this live balloon launch. So that will be the exciting finale. But of course, it won't end there either, because we have to track this thing and see where it goes. So with that being said, I'd like to introduce our student co-leaders, Simran and Karina, who I hope both of them have joined the chat already, the call already. Girls, are you there?
Yeah.
All right. Hey, I'll hand it over to you all.
Hello, my name is Simran. I'm in 10th grade and I go to Crescenta Valley High School. Karina.
I am Karina and I'm 10 years old and I go to Robert Hill Lane Elementary School and I love STEM.
Oh, and actually, before we start, I can't believe I forgot this, but I really want to share a short clip, because we were actually on the news last night.
Oh. Oh, that little thing.
That little thing, yes. I don't have cable, so I couldn't watch it live, but I did get the playback video, and I would like to play it for you all.
Oh, thank you. I haven't seen it yet.
Oh, great.
Wait, no.
Oh my gosh, how do I do
It
this?
is superb.
So
Okay.
do you
Just
screen share?
spoiler.
How do I make it? Why do I sound like I don't know what I'm doing? All right, I'm setting it up. We're almost there. Okay, I'm gonna share my screen.
You got it.
This looks right. Okay, you guys can see the video.
Oh, yeah.
Correct? Okay. Let me rewind to, it's very short. Oh, can you
No
hear
audio.
it though?
No.
How do I share the audio?
Well, the audio should work automatically, but I'm wondering, is because you have your headphones on? No, that wouldn't make a difference, would it?
Probably.
The audio needs to be turned up. Because that happens to me. Unplug the
Check
cable.
to make sure the mute on YouTube app.
I don't think. Yeah, I'm not sure it's YouTube.
Audio. Did you unplug the cable? Janelle?
Zoom is funny about how it handles the audio between your microphone versus playing a video. So
Try water.
and I think the singing there is from one of our happy students.
thought I saw a notice of a mention, but I didn't open it up today. Media alert. I actually have it here. If you'd like me to share it, it looks like I've got it.
Can you hear me now?
Yep.
Yes.
I can hear you.
When I unplugged my headphones, it switched over my audio.
Okay.
That wasn't my speaker. So let me give that another try.
Okay. Good deal.
Okay. Man.
And Ted can also play it as well, apparently.
Well,
oh, okay.
No, go right ahead, See, since you went through all that.
All right. Here we go. Here's the real deal.
Part of the robotics team at my school. I've been participating for 2 years.
Fifth grader Karina Moorhead admits she misses school and the after-school programs at Pasadena's YWCA. So tonight, she's buzzing about how tomorrow the Y is launching this Mylar science balloon.
So this is what we're putting up in the air. We're looking at a 1 meter wide party balloon.
Karina joining a real-time online community to follow this shiny device.
The balloon will get tracked as it goes, and it uses solar energy to connect to the amateur radios, that's what they're called, below, and then to communicate and transmit its location.
The balloon launch Raise Your Sights is just one way for Pasadena's Y to engage students during the pandemic in partnership with the Bay Area tech company Magnitude.io. And who better to lead the students than a local volunteer who is also a rocket scientist. Janelle Wellens knows how STEM education can help girls soar.
Since our program is normally in person, is after school, and the girls come to our location or we go to their school, we had to find a way to keep our programming going and serve the community. And so we brainstormed.
Wellens says anyone can follow tomorrow's launch at 11 a.m. via the website and the balloon call sign seen right here on the screen. As for her focus,
to get these girls to have this experience as a tipping point for them to become more curious about STEM and want to come back to the Y to get more.
You might say Karina is already sold.
In 3rd grade, I was into astronomy. So now, and I'm transitioning to STEM, it's sort of like a good in between because we're learning a track, and even though it's not outer space, it's still around our atmosphere.
In Pasadena, Beverly White, NBC4 News.
Awesome.
Wow.
What a great piece.
All right.
Congratulations.
And Beverly, it looks like she went right over to your offices in Pasadena. And
I just noticed that.
yeah, she was literally there. She got her outside broadcast truck over to Pasadena, literally for like 5 seconds worth of screen time. That's how amazing, she is as a reporter. She's just fabulous. Wow. I wonder if I'm going to find them at Santa Anita Mall.
And I hope I got your call sign right. That's
You
right.
did. Yeah.
All right. It was a last minute thing. And I was hoping we'd do our little, so Liam and I last night pre-filled the balloon because of the logistics of a live broadcast and anything that might go wrong.
Yes.
Show that just before launch. But we're going to turn it back over to you guys, and we want to hear more about what you've learned and maybe some questions we have from our group, whether it's on YouTube. If you've got questions about launching balloons or even the Leguminaut challenge, please make sure you're putting your questions in there. And of course, any of our teachers or guests here inside Zoom.
And just so everyone knows, Beverly is going to also stream the launch of the balloon and this Zoom call. We're giving her the information now. That's what I was texting about earlier. So
No,
now we can
gosh, no pressure.
we can hand it off to our co-leaders. You can take it away.
Oh, there's the balloon in my office, by the way, right now.
Karina? Simran?
Yeah. I'm sharing my screen, 1 second.
Oh, okay.
So this is the balloon launch project.
Powered by the YWCA, Magnitude, and ISS above. So the balloon that we'll be launching at about maybe 11:30, 12 is made out of mylar, about a meter in diameter, I was told, and it will reach the altitude of 24 to 30,000 feet, 7,300 to 9,200 meters. Its direction of travel will be affected by the weather and the wind patterns. The balloon could also be affected by its other surroundings too. And this is a picture Janelle showed us as an example of the probably material of the balloon.
So we will be attaching this Pico Hub to the Sky Tracker to our Pico Hub, and it transmits our position to the amateur radio tracking system every few minutes. So we'll know exactly where this balloon will end up. This link will help us track where our balloon is, and also it uses solar panel, so we can't track this balloon in the nighttime when there's no sunlight. Karina?
Oh, you're muted, Karina.
Yeah.
There you go.
So the weather today is going to be about 73 degrees Fahrenheit, and we put a picture of the predictions basically we got from weather.com about of what the weather is going to be like, and just to also show how how the direction will change probably even with the weather patterns, because the weather is always changing. And the launch location will be the balloon will be launched at the Santa Anita Mall, and it will be the coordinates are 34.1372. I believe that stands for north, and 118.521. I believe that's west, about from latitude and longitude.
So the weather patterns do affect where our balloon will end up, as Karina already mentioned. So these are so we use this to predict like where our balloon would approximately end up after 7 days. So these are the wind patterns.
And so Simran, I had a question about that. This is, can you talk a little bit more about what you were just showing?
This?
Yeah, so you have, there's different layers of the atmosphere, right?
Yes, so the balloon will be at 250 HPA. So, which is different from ground level weather patterns. So, being higher in the air will affect how fast and how much it moves.
Because the higher you are, closer to the higher you are, the pressure is not going to be as much as where you're lower. There's more pressure.
Great, thank you.
So these are the predicted paths of our Pico Hub. This was predicted on May 29th. Now we did another prediction this morning, and it changed so much.
Yeah.
It changes very significantly, and this was made today. The prediction was at 10:19 a.m. and this shows you just how much weather changes in just a few days. So that's also another thing saying to the weather patterns that the weather and the temperatures probably will also affect it. So that's probably most likely not the exact course because of it just changes in a few days. Just imagine how much it would change in 1 week.
And when we look at that graph, can you describe what that red line? I see some triangles on that. Do you know what those?
Oh, the triangles, the big triangles are per day, and the small triangles are the predicted location in every 6 hours. So,
Where does that look like it's ending up in a week? It's going to be, where is that?
Like in
Close to Portugal.
You're heading right to the Mediterranean, I guess, huh?
Yeah.
Oh.
Great predictions.
And here's the link also to, if you want to just go to it, you could just click on the link and you can go to the link.
Now that link is kind of complicated, isn't it, Karina? It's got a bunch of calculations on it. You guys went through that?
Yeah, we went through it.
Mm-hmm.
So these are the coordinates of our launching location. And
I think it's supposed to be the eye thingy. Is it? Looks like that, sorry.
And so there are a lot of variables that you can, and I see. And your source is NOAA. Do you guys know what that stands for?
Let me think.
Pop quiz.
I remember.
it's about the same prediction right now.
Mhm.
We just did it an hour ago.
Okay, cool. Yeah, NOAA is the National Oceanographic and Atmospheric Administration. So they're studying the ocean and the atmosphere. And if you think about planetary scientists, I don't know if C's on with us, but C works with us, and she graduated from UC Berkeley as a planetary scientist. And there's a lot of folks that are interested in exoplanets, planets beyond Earth, in different solar systems and different galaxies. But we got a really special planet we can observe right here. So if you're interested in astronomy and planetary science,
I did when I was in 3rd grade, I definitely did a solar system project.
Fantastic.
I was into astronomy and kind of, I still like astronomy, but like done better a little.
Good.
Yeah, this is the end of our presentation. So.
Well, fantastic. I'd like to open it up here to our guests in Zoom, and I'm going to check YouTube, but I'm sure you have some questions for our young scientists here and our young engineers.
I have a question. What is the difference between the balloon you send or you're sending and the balloons that we commercially buy in the dollar store or something like that?
Well, probably the difference is that it's probably going to be a bigger balloon, or maybe the material. I'm not exactly sure. We haven't exactly seen the exact model of what it's going to be, or just a replica. But probably the difference is it's probably going to also have more helium, which will cause it to go higher, and then also it will probably have differences or different attributes, because a bit's going to go all around. Your normal balloon would probably just go up, 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 and doesn't have as much movement probably, and just pops easier.
I like that hypothesis.
Yeah.
I guess,
of course, as I'm speaking, you can see your balloon is on my ceiling right now.
Yeah, girls, there's with Liam's shot there. I mean, why does your balloon kind of deflated? It's not like nice and puffy like we usually see.
do you have an answer for that? Or it's kind of a little bit of we need usually a little bit more time to kind of dive into these investigations, but your clue to your answer is back to what you were talking about, pressure and how pressure changes in the atmosphere. So we've got a certain amount of gas, helium gas, inside that Mylar balloon here on Earth, and there's higher pressure here on Earth, right? And as that balloon goes up, you told me, Karina, that we're going to get less pressure, right?
Yes.
So what's going to happen to that volume of gas inside that balloon when we have less pressure on the outside?
It's probably going to grow bigger.
Yeah, exactly right. And so I guess we
they're not so close together.
Exactly. And so if we pump it up full of helium here on the Earth, it's going to probably go up faster. But what might happen? You ever fill up a cup with too much of a liquid? It's going to start spilling over. Your balloon might burst, right? Because it's going to have too much pressure, and there's not enough pressure on the outside to hold the balloon together. And so I think our answer has something to do with that pressure.
So the buoyancy has to be really carefully calculated then. You can't just randomly throw some helium in there.
Oh my goodness. Is that a good prelude for our introduction video, Liam?
Yeah, sure. Our 2-hour journey last night.
Well, I've got it down to an hour, 1 minute and 44 seconds. If
That's
I can.
right. Thank goodness. You don't have to be with us for 2 hours.
It's only just, well, why was that? It's interesting. It shouldn't take that long, but we were both learning together and trying to do it remotely. So,
yeah, I was like the remote hands of a remote operated robot.
little telesurgery going on there.
Yeah, the operator was in Berkeley, and I was down, imperfectly following instructions.
Well, and I think what it was is the person giving instructions just was kind of lame. I was giving you the best instruction. The thing.
So, Liam, you can no longer check the checkbox on CAPTCHA. You are a robot now.
Yeah, I am definitely a robot.
Oh, there you go. All right, so let me share my screen.
I'm going to bump over to YouTube with my students, so we'll hopefully be able to watch this launch.
Yeah.
Have good luck with it, and catch you all on the other side.
Have a good class, Michael. Thank you.
All right.
All right. So here's Liam with a summary and a good overview of what it takes. And normally, we're working with you guys directly. You guys get to do this, and on our bigger balloons, you actually even contact the Federal Aviation Administration to file a notice to airmen because those big balloons kind of get in the way of stuff. This little balloon we don't have to worry about. But I think we're doing the best job we can, considering that we've never really met in person. This is pretty cool. And so I really have to thank Janelle and Jomi and Cynthia for their leadership at YWCA and giving you guys this amazing opportunity. Really excited about this. And I will share that this is a prelude because we have some other teachers in the Central Valley in Northern California that will be doing this every week. And we're leading up to a balloon that's going to circumnavigate the planet. Different type of balloon. It's going to be a great mission. It's going to happen probably within the next month. So definitely stay tuned. But here's Liam giving us a nice summary after a couple hours of work of what it takes to put this together.
So the recap is it's done. The balloon is assembled, inflated, configured to exactly what it needs in order to, when we release it tomorrow, to launch up into the sky over the Santa Anita Mall and heading east towards the San Gabriel Mountains. It really is, just a matter of follow the instructions, work out all of the weights that need to simulate the weight of the sky tracker as you're filling in the helium. I can't tell you how many times we filled it up, got it so that it was looking like it was the right lift, and then it was a little bit too much, so I hugged it a little bit, and then got it at the right level. And just so as you know, yeah, we sealed everything up, did it again, and it wasn't quite the right lift. So we had to backtrack and disassemble some of the tape that we put around, and then put a little bit more helium in. And we think that was due to the weight of this Starbucks straw in comparison to the official white straw that is supposed to come with the kit, but just didn't anyway. But the end result is it's all safely there. It looks like it's well connected, and it should be, very well set up for the final launch tomorrow and for a safe ride across the United States.
Now you can see I ran that model last night, and that's different than the model you just shared this morning. Mine also had 3 different colors on it because I was unsure of the altitude. And when you go back and do your calculations, if you want to, again, ladies, what you can do is you can actually put in different heights to see what might happen if the balloon's a lower altitude. Is it going to go a different place? And it's going to do it a little bit. And as it goes farther, several days into the future, you're going to see there's even a greater disparity in terms of altitude change and what have you. So that was kind of neat. But Liam, thank you for your patience of going through that. And something that Liam and I didn't think about, it's like, oh, we need to infill this thing, but this is the normal straw or filling a pipe that Bill Brown that makes the Sky Tractor provides. but I didn't have one to send Liam when I sent him one back then when he received it. And so he just grabbed another straw, but there is a difference in weight. This thing's about twice as long as that straw. It's a much thicker material, and we didn't calculate it. It's like maybe a half a gram. And so at the very end of that video, you could see Liam attached a nickel to his balloon. A nickel weighs about 5 grams. And after we did our calculations, 5 grams would make that balloon sink to the ground. Just that little bit of weight, just the weight of a nickel makes that balloon go down. But if we added a penny to it, Liam added a penny, it was supposed to go up, because a penny weighs about half that amount, about 2.5 grams. So really nice, convenient weight system there just by using some coins. So, but we attached the penny to it, and Liam, that thing started going down, and we were like, oh, first of all, he had it all wrapped up and it was all ready to go. And it's like, we did all the math, it should work, but we didn't think that he had a different straw with a different weight. Something so subtle as that made the balloon drop down. So we fixed that, and it looks like it's doing everything it's supposed to do now. So that was kind of an adventure, to say the least. It took twice as long as it probably should have because of that.
Oh, it was a fun time though. No regrets here. So I'm thinking I should probably get in my car with this balloon and sort of, drive over to the Santa Anita Mall.
There you go. Now, it's about a 5-minute drive for you. Is that right?
Yeah, it's 12 minutes according to Waze. So yeah, so I'll go, and I'll get onto the Zoom, when I'm on my way over there.
Oh, there you go. Yeah,
So,
drive safe and pop in. We'll be chatting about different things and keeping everyone entertained and informed, and probably having some curious questions come up too. So
cool. And I definitely want to say to all the students, you did a fantastic presentation there. I'm so proud of what you took on and how you presented the whole sort of aspect of what's going on here. So you did a great job, and I can't wait to launch this on your behalf in just a few minutes. So that'll be very good. And Karina, we must make sure that I get my astronomy crowd here to come to your school. That's my specialty too. So very good.
About the material of the balloon exactly. So like you said, that if it has too much helium as it goes up, it will pop. Well, what if it's going over the ocean? Will this hurt the animals? Because we also got to take other factors into consideration. Even though it's our experience, we also don't want to hurt the animals, and not being able to pick up trash right now is kind of harmful. And I was in Eco Club, so this is also why it's a concern. But then also, I just want to try to have this being eco-friendly.
That's a very important question to ask. I'm glad you brought that up. So there is the Mylar balloon. It's traveling with the winds. We don't know where it's going to go or where it will land. We hope that it lands here on Earth where someone could recover it. But there is a chance. I mean, there's more ocean than there is land. So there is a chance it does land in the ocean. If you could find, as a inventor, a creative way to run atmospheric experiments with a biodegradable balloon, I would be so excited to fly one of those. Because that means it would be, it would just break down in the natural kind of chemistry of things. But anything that's biodegradable like that won't last very long, so we can't get those long flights. It's a fairly small balloon, but still, I guess if we think about it, we have to clean up after ourselves, right? Underneath those solar panels, there is some information for anyone to contact us. So if it does land in the desert somewhere or in a mountain, and someone comes across it, they can contact us, and we can get it back, which means we could fly it again, which would be pretty cool. Our really big balloons, we fly on latex balloons, just like your balloon you might get at a party store, what have you. They get really, really big, 3-4 meters in diameter. We fill them up with a whole bunch more helium. They go up a lot higher, but they don't go nearly as far. But what I like about those, two things I like about those balloons. First of all, it involves getting in and doing some engineering. What do you want to put in your payload? Right now, you didn't really have a chance to do much engineering with this. You're learning about the radio, and you're learning about solar power, and you're learning about other things like GPS. But you're not constructing anything. With our larger balloons, you actually get to design and construct things. We have some students that put like plants or seeds in there because they want to know what happens if we send seeds past the ozone layer, because those big balloons go up about 100,000 feet, 3 times higher than a commercial airliner. And then Tony, I don't know if you want to pull up a quick little video showing one of our balloon launches, maybe the one with Vertibi School of Engineering. This was another girls program there in USC, so it was in your backyard. But that was with a much bigger balloon. Janelle helped us along with that, as well as another teacher, Christine Hurst. She works at a high school and she does balloon launches all the time. She's really pretty good at it. But these balloons go up, and when they burst, we can get them back. And so we don't worry about what kind of impact we have, because we can recover them. But I'm so glad you brought that question up, because I have a little bit of guilt when we launch these because I'm unsure where it might land. And so if we can find a way where we can get these long distance balloons going and not worry about that, I would absolutely adore a solution. So think about that. Simran, Karina, your friends. I think I saw Ura's here. You guys could figure out a way to do that. I'd love to try. So here, watch this video. This is pretty cool. This was done not too long ago, a couple months ago, pre-pandemic, for sure.
Catalina with USC. We're here today with Project Payload. So we have a couple teams working today preparing for their weather balloon launch.
Why do you think this wave is going up and down like that? What's happening? We want to see what it looks like, how much helium we need in order to go, what our ascent rate is. So we're actually going to calculate this now, but we got to do a weigh-in.
If you give yourself the chance to take the harder classes, take classes that you may not be that great at or may not like as much, at least you're giving yourself the opportunity to do anything. You're not limiting yourself. So the more you think on that kind of terms, the better off you'll be. Trust me when I say fine. And my mom calls me and she's like, you need to get home right now. I was like, what happened? What? She's like, you have package to out from MIT. I was like, Mom, I already told you this. I didn't get in so you can relax. She's like, well, get home now. Waiting at the door. She's like, here, handful packaging. I'm like, Mom, what are you getting excited about? You're just telling me no. She's like, Janelle, did not send you a package to tell you no. Good point. I open it up and I read the first letter that says, congratulations.
I don't know how to use that one.
My name is Summer. I'm the record keeper, so I have to make observations of what I'm doing. Right now, and the balloon team. So the payload team is right now putting an egg, figuring out how they can find the egg so it won't crack when we launch it into space. And then this team right here on the right of me, they're figuring out ideas how to tie the balloons and tie the knots for the balloon, and so how it can expand.
Hi, I'm Roshan. I'm one of the instructors in the program designers for Project Payload. I'm a student at USC, and we work with the K-12 STEM outreach programs at USC Vertibi School of Engineering. This is Project Payload, and we have over 20 middle school students. They're all girls joining us here with Project Payload to have a program that helps them go into the STEM fields and learn about a lot of things related to STEM. In our project, it's been going on since August. We've had a 2-week summer camp where they learn about programming, Arduinos, cube satellites, how they're launched, how it works, leadership programs, how to better communicate with one another. And throughout these past months, we've been having sessions every other Saturday to learn about cube satellites, CanSats. Right now we're working with Magnitude so that we can launch a high altitude balloon into the upper atmosphere with a helium filled balloon. And yeah, it's really exciting.
Wait, are we letting it go on one?
No, I'm gonna hold it.
I guess Carl the balloon had a birthday. It's gonna do a little. You reckon? Yeah. That was a little bit of a story.
That was the coast circles right there. I know, right? What a great story! Right? That is awesome. So this is, there are some that pretty much keep into the radar, don't report anything, don't tell anyone what to do. The other one is, for me, let everyone know what you're doing because you're doing something pretty significant, and they want to help. We're here at Dana Point, where the Orange County Sheriff Harbor Patrol, the boat that was recovered, the high altitude balloon that went to 100,000 feet. You guys are heroes, man! This is Ted and Toby. Very happy. Oh my goodness, we have some young girls that are super excited at USC. They were working on their payload. They got it up. We were thinking it was going to land right there on the coast. We didn't think it was going to go in the water. County Sheriff, big hand here, making a dream come true. Look how salty it is.
Isn't that fabulous? Now, see, what's nice about those big balloons is we get the hands on and we get it back, right? And this one was, oh my goodness. So we ran the model, just like you guys ran your model, but we have to recover the big balloons. And the morning it looked like it was going to go into the ocean. So we waited like 2 hours and we ran the model again. And it looked like it was going to land on the coast. So we said, let's launch because everyone's ready to do it. We launch it, and about an hour into the flight, flights are usually about 3 hours long with the big balloons, sometimes a little more, sometimes a little less. And about an hour into the flight, you could see that it was doing something we really didn't want it to do. It was starting to move toward the coast. Now, with those big balloons, we have to contact the Federal Aviation Administration. We basically file a notice to airmen. We let them know what time we're going to launch. We let them know the direction we're heading in and our approximate altitude, other kind of calculations. Because we're in an urban area, we usually contact county sheriff or local municipality, the government, to make sure they know that something's happening. Our thought was if it landed in a gated community, it would be, you can't just jump the fence. So making sure that everyone knew that we're about to do this was important. So here, an hour into the flight, we're like, oh, we better call the sheriff. And they already knew about it. So we contacted the watch commander and we said, don't have to worry about our balloon. It's going into the ocean. And of course, that's making us sad. And we're worried about our animals in the sea, right? Our fish and what have you. And we didn't want that to be in the water. But we're kind of disappointed that this is what's going to happen. And the watch commander says, no problem. I'll call Harbor Patrol. I didn't know the county sheriff, Orange County Sheriff, had a harbor patrol. But literally, within 30 minutes, they dispatched that boat that you saw. And that balloon landed in the water about a mile offshore. We couldn't even see it. We were on the beach. We were trying to look. We didn't have our binoculars. Probably should have had some binoculars, but we couldn't see anything at all. But I get a phone call. I pick it up, and it's the dispatcher at the sheriff's office saying, they just picked up your balloon. Literally, it was in the water for about 10 minutes. It really wasn't wet inside at all. It's a little damp, of course, on the outside, but wow, how awesome was that? Now, we don't want the balloon to go in the water, and we try to run our predictions, but that was one of our more unusual experiences. So once all this gets passed, and if you guys are really interested in these kind of projects, working with Liam and with Janelle and YWCA, maybe we can make a plan to do a big balloon launch in the near future, which would be really pretty fun. But let's focus on this right now. Do you guys have any questions about like that big balloon versus your Mylar balloon, which is going to go much farther? Your balloon's going to go for days. Again, ours, that big one stays aloft for maybe 3 or 4 hours.
So I have a question. So the balloon's going to travel, and what has happened with many of the balloons? What has happened with them? Where have they ended up?
That's a great question. So we launch them from different locations. Quite often we're launching them from Northern California. We had one balloon that went down, well, the one we launched last week went down into Mexico. And if we look at the ground stations in Mexico, there aren't many. So I think there were some near Monterey, the state of Monterey. And I think there was one right at the tip of the Yucatan Peninsula. And our model looked, it was going to fly down in that direction, but we haven't heard anything of it since then. And our model, if we go rewind all the way back to the last time we heard from the balloon, if the model is correct, it's currently halfway over the Atlantic somewhere. Now, we've had the longest distance flight from California. Our last blip we received from that was off the coast of Africa. There's a little island chain called the Azores, and that was a blip we got from the radio station that was there, and it was flying right over Africa. So it's probably in the desert somewhere, and some sand dunes, and some guy on a camel is going to find it and is going to go, "What's this?" Right? We had another one we launched from Kentucky, so that's on the East Coast, and that one we saw it going up towards Nova Scotia near Canada, and it went over the North Atlantic, and it disappeared for a couple days because, again, there's no radio towers in the middle of the Atlantic, and then it woke up coming down near Britain on the way to France, and it came down through Spain, and then we lost it again because it went down into North Africa. And we're like, oh man, we lost another one in the desert. And then like 2 days later, it woke up again, it showed up, and it was going in a different direction. It was heading into Greece. It's like, wow, okay, it's back again. And it flew for the next day and a half. I think we watched it and we lost it in Bulgaria on the way to St. Petersburg, Russia. Now, if it kept on going, the map said it was going to go around Mongolia and come down through China and then back out through Japan. These balloons are unlikely the Mylar balloon will go as far as around the world. I mean, it possibly could, but usually they don't last that long. But so far, we haven't seen any indication that they've landed in the ocean per se. Now, the balloon, I just got them yesterday. These super pressure balloons, we're going to launch in about a month. They go up higher than the one we're going to launch today, but lower than those big ones you just saw. They'll go up to around 45 or 50,000 feet or so, and they will go around the world if we pick the right weather, if we got the right day, the wind pattern looks good. That Earth.null school, that great visualization you guys showed us earlier with the wind patterns, if those wind patterns are just right, we can go all the way around the Earth, which would be really neat.
How come you haven't used those ones before? Have they barely been like available? Or are you testing out different types of balloons?
So these balloons are normally done by researchers in, like, if you're a graduate in university, you're studying the atmospheric investigations. At major airports, every morning, they usually put aloft a high altitude balloon, and the instrument they put on it, they give it a name. It's called a radiosonde. And it's very similar to what we have in classrooms. We use something called a CanSat, which is a simulated satellite. So in the volume of, imagine this is like a soda can. It's a little different, doesn't really look like a soda can, but it's the same diameter. I've got half of it here. Yeah, here's one, here's the full one. So, here'd be like a soda can equivalent, 122 millimeters in length and 65 millimeter diameter. But inside is a bunch of electronics. And so our different balloons, we run different instruments, and that radiosonde that launches from the airport measures things like a temperature. We have a temperature sensor and a humidity sensor on this. It measures things like barometric pressure. And of course, we know that pressure gets lighter the higher we go in the atmosphere. We can actually calculate altitude just from the pressure sensor. Here's a super big GPS block, and it's about maybe 8 times larger than the GPS block that's on the balloon that Liam's about to launch, which is kind of neat. And it looks like Liam's in the parking lot right now. And it has other sensors on it as well. And in addition to that, a real-time clock and a data card. And it's running on an Arduino. I think you guys have done some programming before, code.org or what have you. So this is the Arduino microcontroller. It's called an Uno, and they're really inexpensive. They're like 20 bucks, 25 bucks. And you can program it. You program this little chip. It's called an Atmel chip with a set of instructions. You tell it to turn a light on or turn a light off, or if the temperature gets too high, turn a fan on. In this case, we're just saying, would you record a bunch of instruments, a bunch of sensors for us so we can measure the world around us? So this electronics is kind of like the human body. You know, we got eyes we can hear, we got ears we can, eyes we can see, ears we can hear, we can smell and taste and touch. And this is doing some electronic versions of that for us here. So if you're interested in those sciences, we can do it. We launch rockets sometimes too. But I really like and I appreciate your concern about pollution. And I think that is a great path of discovery. Can we find a biodegradable balloon that we can do research on? That would be fascinating to maybe try some different things. I know in like ancient cultures, like in China and Japan, they might just do heat and they run their lamps, right? And so they build a lantern, and just the heat of the candle will lift the little paper lantern up. It's a fire hazard. You got to be careful about that. But the idea is that you can watch those going up at night, and they look like these little stars as they go up. And that's, if you look some history, just by using hot air expands, and so it rises. It's not as efficient as helium or hydrogen gas, but that is biodegradable, especially if it's a paper lantern, right? But you got to worry about the fire. All right, hey, so I'm going to turn it back over to Liam. I guess you might be on mute. Liam, what's it like out there? Like you're
just take. Can you hear me? Just check audio check. Check.
We're in.
Yeah, we can hear you.
Good. Yeah. So I made it to the mall just to prove just how empty this parking lot is. Let me just go around here. That's this is the Santa Anita Mall. Say hi, Anna.
Oh.
Anna says hi.
Anna, thank you so much for helping last night. Your extra set of hands made it worthwhile. We were able to get it done.
Yeah, and Anna can't hear. I've got you on my Bluetooth headset, but I'll definitely relay that. And then over this way over here is obviously the Santa Anita Mountain.
So you're looking north?
Yeah, that is towards the east.
Oh, east.
So that should be the direction that the balloon is going to be heading. And I also have over here my ham radio. You know, and I'm picking up all of the local radio stations that are transmitting that's on the same frequency as our PicoHab balloon. Now our balloon is just in the back of the car right now, and it's currently out of the sunlight. So, you know, what does that mean if it's not really in the direct sun? Hello there, how are you? Oh, yeah. So we're just doing a quick balloon launch that's going to be on NBC News. So we'll be leaving just in 5 minutes, if that's okay. Okay. All right. So it looks like we've got a little bit of a problem here to do this. We're broadcasting this live at the moment. So sorry for. Yeah, we thought that it was good because it was open for food. Oh, say that again. Okay. All right. Yeah. So this is what happens with.
So put it up right now, Liam.
Yeah. No, they literally are telling me I can't launch it from here. So they're saying I have to get off the site. So I'm going to go over to the parking lot of the Arboretum, and I'll be right over there.
Okay. My goodness. Thank you for being a good citizen. I would have actually just put it up myself. I'm a naughty boy.
Yeah. Okay. Well, not with them right here. They're actually waiting for me to get out of here.
Okay. Good deal. All right. So go ahead and work your way over there. And we've got time, and it's okay if we're a few minutes over.
That's fine. Yeah. We're just gonna.
So is he launching from different coordinates now?
Yes, but you know what? I think it's probably going to be all right, because remember you had your latitude, and you had like 4 decimal places. It's like those last 2 decimals are going to move around.
So it could range to like the 2 or 1 could range from that.
Yeah. Now, if you wanted to, if you want to get super accurate, and I really like that, if you want to do it, ask Liam well, he said he's going to the Arboretum.
Oh, wait till he gets there.
Yeah. Or if you look at the map, pull up like where we can do that right now. Why don't I share and let's do it together. All right. This is cool. Science. All right. A little engineering. So maps.google.com.
What a twist.
No, I know, right? There's always a little adventure somewhere. And of course, she's got to show up like right now, right?
Right as we're about to
Liam said he was going to bring donuts for the cops, and because he saw cops last time, but he didn't bring donuts.
Yeah. Well, and just let you know, yeah, the last time we were here for about 10 minutes, and no one came over to us. So there must be just a slightly changed priority in terms of what they're doing, and this is open to pick up food, so which is the rather unusual thing. Anyway, where I'm going, because we can't actually go onto the property of the Arboretum, but just a minute from here, it is their parking area, and I've just got to hope that I'm not going to encounter some. Just make sure I'm not going to encounter some trees, because we're heading to an area where there's a little bit more trees. So, and you know where this is, Tony, because you've been to the Arboretum here. Yeah, this is a bit of an unexpected adventure. So, but here we go.
All right.
Yeah, here we go. So I'm driving into the parking area of the LA Arboretum. It looks like, yeah, I'm going to try it just over here.
All righty. And so I'm going to just put.
Yeah, we got peacocks here.
Oh, I think I might know the Arboretum. Is it like in Arcadia?
Yeah.
Okay, so I pulled up the map, Liam, and so Liam was in the parking lot over here, right? And he's now moved over to over here. And I'm wondering a parking lot. So there's that little roundabout there. You're not there. You're kind of near the entrance of the Arboretum itself.
I'm going to try and, oh, there's a bus right in my way to do a U-turn here.
No rush. We're totally fine on time. NASA's just added 10 minutes to our flight clock, I think.
Yeah, there you go.
This is not a simultaneous launch. We don't have to get orbital.
Yeah, I'm curious whether NBC is covering the live stream right now.
Oh, there you go. And you were being a good citizen on top of that.
So they'll, here we go. All right, so I'm just going to head around here and head into the parking lot.
And so
empty.
And I know you're busy now, but I'm curious if we can just get a sense of where you might be on the lot, and then we can pull up a quick lat and long.
Yeah.
We want to run a new calculation.
Yeah, so the main entrance to the Arboretum is about, I don't know, 300 yards from where I am. I'm slightly to the south, probably southwest-ish.
Okay. So
yeah,
there's
so see where that fountain is. I am, I'm relatively close to that.
Got it. But you're south, so you're south of the Arboretum entrance. So he must be down here.
Oh, hang on. Yeah, let me. Yeah. Okay.
Just you.
Sorry about this.
That's all right.
All right. Can you hear the peacocks? You might not be able to. They're very loud.
Hear them.
Very loud peacocks. All right. So here we are, and yeah, I think we're just going to do it here.
Yeah.
Yeah,
Liam, I worry about your ascent rate near those trees. So
yeah,
go up as fast as those larger halves.
Right. So I can feel the wind appears to be going, towards those trees.
I know.
Yeah. So what do you think? Do you think I should try and find somewhere else?
Well, it's hard for me to judge from there if I don't see a windsock or anything else that gives me wind indication.
Yeah, it's very low wind.
Okay, well that's great. No wind would be ideal.
But it is, well, this is its own windsock, right? Can you see the balloon?
Well, there you go.
So you being the expert balloon launcher, what do you think?
You're gonna put it on me, huh? Let's take a vote here of our young engineers and scientists. do you think we should try a different location? Different that would change our time, which isn't a big deal. Or do we think this might be worthwhile? We've got, I guess I'm going to try to count parking lot spaces there. You've got about 10 car lengths, or what, 6 car lengths before you get to that tree I'm seeing?
A little bit more than that. Yeah, it's a little bit further away than that.
Yeah, I think you're.
The balloon usually rise. And that's
That's a great question. And you know, I didn't calculate the lift in terms of lift per second, feet or meters per second. As you recall in your living room, Liam, it wasn't lifting up very quickly, right?
No, because I don't think it's supposed to, right?
Right. And so you've got in this little trigonometry here, you've got the angle of lift and you've got the distance, right?
Mm-hmm.
Now your angle of lift, you're going to have an acute angle there. It's not going to be like 45 degrees or 90 degrees because you've got some wind. Very high wind, it's going to be very low angle, which means you have a chance of running into those trees. So literally we'd have to do some math calculations.
It looks like it's heading over this way.
Okay.
So the wind is taking us away from the nearest largest trees, and we can get further back.
Okay. Yeah, the further back you can get, I'd recommend.
so it gives me more time to lift, and then after it just gives a better balance for everything.
Other things we could do if we were concerned about that would obviously change the location or change the time, or even change the device itself, adding more helium, which means you may not get a long flight, or lightening your payload, which means
now the wind has died down. This is good.
Yeah, and it looks like you've got no wind. How about a countdown, guys?
Do you want a countdown?
Yeah.
All right.
Yep. Let's do it. All right. 10, 9, 8, 7, 6, 5,
4, 3,
2,
1. Go! All right, that's there it goes. That looks to me, it's going to be clear of the trees.
Yes. I saw that it's going to look like no wind. I said, let's not wait. Let's just do it. And you're about 90 seconds over our midday launch. So I'd say that's a really good timing, despite being set back from the security folks. And we're aloft. Now, our calculations would be slightly different, right? But I wouldn't worry too much about that difference. And Liam, just out of our confidence, can you grab us a Latin long right there? Our gals might want to do a calculation anyway.
Yeah, I can't do it because I'm here, but anyone can just do. I mean, I can definitely let you know precisely where it is, but it's going to be within the margin of error.
But sometime here midday, you just send a note out with the Latin long. Time, it looks like we launched around 12:02. That's our time of launch. And I'm not sure of the altitude there at the Arboretum, but if you want to be really accurate, then that's the launch altitude.
Yeah, I don't think that goes into the model though, right? Does it? Karina, yes.
You had a question?
Oh, so there's just something wrong with the audio for a second.
Oh, okay.
All right, and also I want
location. Can I have the exact coordinates? I wanna know the coordinates. Sorry.
Yeah.
And I'm very
I can give you an estimate.
Sorry. Yeah, I wasn't hearing.
I'm going to share my screen. And Karina, I'll show you what I think, and it's going to be an estimate, but based on where I'm looking at their geography, the trees around them and whatnot, you see my screen right there? I think this is the big tree that we saw in the picture. And there's that palm tree, right, with the shadow. I think Anna and Liam were back here when they launched, right? So that's just a guess, but your latitude and longitude would be, your latitude is 34.1422, okay? And north, and your longitude will be 118.0521 west. 34.1422 north, and then 118.0521 west. Plot that in. You can put your launch time at, just go ahead and make it noon. I think they don't give you minutes there on your calculator.
Same location. Let me just check the previous ones where we had it. I have it written down. The
Okay.
previous ones was 34.1372, and I believe that was the north one possibly, and
Yep.
after the one on 8.0521 was the other one. So it's in the same numbers of the whole numbers.
That's interesting. So the latitude seems like it was the same. So as we go from west to east around the Earth, you were in the same latitude, but your longitude was a little different, right? So it was a little further north than where we originally were planning to launch. So not a lot of difference. Clearly, it's probably maybe a quarter mile or half mile here on Earth, but Earth is pretty big, so not that much of a difference. Can you still see it, Liam?
Yeah, it's pretty high up right now. Is anyone checking APRS?
I'm going to log in right now. I was just checking for the
presentation.
It's still showing it at your home.
Yeah, so it hasn't picked it up yet then. So, and I am seeing if I'm picking it up on my own unit here, my ham radio that's on here.
And can you shout out your call sign again for me?
KN6EQU-1.
Cool. Yep. So
Yeah,
we'll
there's a lot of traffic on the local APRS here, so
it might take a few minutes. Fantastic.
KN6, I've seen it. I've seen it.
Okay, it just popped in.
EQU, yeah, on my local, and it currently says
the
altitude
it's showing it on APRS now.
Yeah, there you go. So I got that on my local APRS on my ham radio here. So that's I'm actually receiving that direct from the unit that's above there. It says it's 0.13 miles away from where I am standing, but obviously most of that is up. And I
There you
cannot
go.
still see it. I cannot see it.
Yeah. Will it like show all the movement recorded where it'll go, right?
It will. If we wait about 5 minutes, it's going to give us another update.
Okay.
Yeah, the wind's really picked up down here now.
Yeah. We got lucky.
Yeah.
We got lucky.
But there we go.
All right. So follow along. You've got the website and the address, aprs.fi. And Liam's ham radio call sign is KN6EQ1 EQU, I'm sorry, EQU-1. KN6EQU-1. And it's on its way. So we're at a little past the top of the hour. And so we're going to wrap our show up here. Always kind of fun and exciting. We're going to do another one next week back in Northern California. Our students from Lodi Unified School District will join us. And young ladies, track this balloon. I'm looking forward to following it today and tomorrow and hopefully over the next week. And get ready. We'd like to follow up with you guys on Monday, I believe. We're going to find out what this thing has done. And we'll talk a little bit about our high pressure balloon and this idea that we want to circumnavigate the Earth within the next 30 or 40 days. So get ready for that. That's pretty exciting. Thank you for being guests and sharing all your information. Hopefully some folks have learned something new or they're excited to learn, go and explore a little further. And don't hold your questions up. Simran, thank you for your leadership. Any of your gals have questions, bring them through your leader. They'll reach out through Jomi, Cynthia, or Janelle, and we'll make sure we get some answers for you on Monday. We may not have an answer. We might have some more questions, but it's always fun to have that great conversation.
Yes.
Liam, thank you for your leadership there and getting this thing aloft. Anna, thank her. Again, those extra has made us all worthwhile. And for now, here at Magnitude in Berkeley, California, we're going to call it a day. Thank you for joining Magnitude Live episode 10 as we make these things up. If you have an idea you'd like to share, maybe a legume recipe, want to come join us in space on our next experiment with Exolab 8 in February of 2021, go to Magnitude.io Check out the Leguminaut Challenge. There's no cost to get in the early phase 1 trials in which we need to find the right plant to send to space. We need your help. Thank you, everybody. Thanks for joining. And we're going to catch up with you next week.
Bye, everyone.
Bye.
Later. Always fun.
All right. Very cool. But I might just do a local shout out on the local ham radio next year, just to let people know. So let me just switch over to my. All right. Let's just see.
Signing off.